Okay, so today you're going to learn all about footprinting, um, sort of the latest advances in the method, various ways to do footprinting, and we're ending the day with a practical session so you can learn how to start doing the footprinting data analysis. So by the end of this workshop, you should have a handle on the concepts behind footprinting, how to collect footprinting data, and hopefully have started learning a little bit about how to do the data analysis in footprinting. So we have a lineup of really terrific speakers today who are going to give you all the ingredients so you can learn all about footprinting and how to do it, and hopefully apply it to your own interesting biological system that you're working on. So I'm going to give a brief overview before we start. So footprinting is a structural biology method, gives you really valuable information about protein structure, protein dynamics, protein-ligand interaction, protein-protein interactions. It's really not as well known as crystallography or small angle scattering, NMR, cryo-EM. And so as I was preparing for this workshop, I started to wonder, how not well known is it really? So the first thing I did is I looked at the protein data bank, and I plotted out the number of crystallography structures that are released every year. And it's quite high, and it's probably no surprise to anybody. So some years up to 10,000 crystallography structures. There's fewer NMR structures in the protein data bank, of course. And there's fewer EM structures, although, as we all know, that's a rapidly becoming popular method that's had some really great advances in the last few years. So I suspect that'll go shooting up. But the protein data bank is really not the right place to look to see how well footprinting is doing, because with footprinting you don't get coordinates that you can deposit into a data bank. So then I looked at PubMed and just did a really quick search with the keyword protein crystallography to see how many publications there are per year with this term, this keyword. And you see there's quite a few. There's a couple thousand every year publications that use that term, protein crystallography. So if you look at oxidative footprinting now, you'll see it's on the order of a couple dozen publications every year. So a couple orders of magnitude less than crystallography publications. So I guess the question is why? Why is this not a well-known and well-used technique, especially considering that it can give you some really good, valuable information? So just in the last decade, of those dozen or so publications, we have some really nice work on membrane proteins. So this is your nice work that Sayan Gupta did a few years back, looking at a zinc transporter protein and the mechanism behind how that metal is transported across a membrane. More recently, there's been some really nice work by the Kobilka group working with Mark Chance's group, looking at GPCR and how it interacts with G protein. There's some really nice work in vivo footprinting methods, and we're going to hear from Lisa Jones later on in the session on latest advances in looking at protein interactions in live cells, which is really quite an interesting application. Speaking of in vivo, Sarah Woodson has done some really great work looking at ribosome assembly, again in vivo. Caroline Aho-Franklin used footprinting to look at a protein binding to a metallic surface. So she was looking at proteins that are on the outside of specific organisms that bind to minerals and then use that as a source of electrons. And Cheryl Kerfield's group has used footprinting to look at these carboxysome, these cellular compartments that are made up of a protein shell. And she used footprinting to look at how the proteins in that shell interact with each other and to determine, use that information to determine how those proteins fit together. So it's really, I think, a really valuable method that can give you really interesting information about any system that you're looking at. And one of the reasons I think it's not as well used as it could be is people just haven't heard of it. It's relatively new. So I sort of think of it as the new kid on the block. But I think that, you know, part of the problem, because it's not well known, is that or one of the results of it not being well known is people aren't trained in it. And so there's a barrier to getting started with footprinting. And so that's really the purpose of a workshop like this, is to try to help people get into the field and be able to use footprinting for their own applications. And my vision is to make footprinting as widely used and as easy to use as crystallography. So hopefully by the time I retire, I'll achieve that goal. So really briefly, how does footprinting work? So for nucleic acid, one of the earliest applications of footprinting was to use some kind of DNase to cleave DNA. But the key thing here is that if you have a protection on the DNA at that particular spot, you block that DNase. So then if you run your fragments out on a gel, you can figure out exactly where that protein was bound to the DNA. So that's sort of footprinting of nucleic acids in a nutshell. Now, a little later on, Tullius's group discovered you could use hydroxyl radicals for the same effect. So you basically can use hydroxyl radicals, which will cleave the DNA backbone, but the key thing is they cleave it at every single base along the backbone. So now if you have some kind of protection on that DNA, say from a protein, you use hydroxyl radicals to cleave it, but you get these larger fragments of the DNA. So you can see very precisely where that protein was binding on the DNA. So again, in this method, typically what people used to do is they run it out on a gel, and you could very clearly see the pattern of protections along the DNA and figure out the specific binding of the protein, specific areas the protein was binding to the DNA. Okay, so with protein footprinting, essentially the same concept. So I'm going to show you a little video of X-ray hydroxyl radical footprinting. So in this method, we use X-rays we irradiate a solution. The X-rays will interact with water and create hydroxyl radicals. And the radicals will modify the side chains of proteins. And one of the key things in this method is that the hydroxyl radicals modify side chains, but only in the local vicinity where the hydroxyl radical was created. So we irradiate our solution, we then take it out of the beam, we add a protease to digest it, and then we run out our samples on mass spec to see where those modifications occurred, those covalent modifications. And from that pattern, we can tell where the water was around the protein. So just in slightly different format here, you can see that we have the modification, we do the digest, we run a mass spec, we look for where those modifications occurred. And just to give you a sense of how this is useful, so say you've got a protein that goes from a compact state to a more elongated state, and you're looking at it in these two different forms. In the compact state, you'll get a modification, say at one particular site, and you'll get that same modification in the elongated state. But now, in the elongated state, you have these additional modifications. And from that pattern, you might be able to infer that that's where the two domains, say, came apart. And so this is how the solvent accessibility information gives you information about structure. So just a couple of things to point out about this method that are really important to note. So first off, the hydroxyl radicals have a lot of different ways of disappearing from solution. So they really don't diffuse far before something else happens to them. So they can modify a side chain, but only if they are right next to that side chain. They will also react with other hydroxyl radicals. They react with water. So they very quickly disappear from solution. And of course, there's lots of scavengers and buffers as well that'll eat them up. So it's important to note, just because if hydroxyl radicals were able to diffuse everywhere in solution, and you use this method, you'd get modification everywhere and you'd get basically no information. And the other thing to note is that the modification depends on both the proximity of the hydroxyl radical to the side chain, but also on the reactivity of that side chain. And there'll be other people in this workshop talking more about those technical details. So you can there's a lot of different ways to generate hydroxyl radicals. There's Fenton chemistry, chemical method. You can use X-rays, other high energy sources, electron, gamma ray. There's UV irradiation of hydrogen peroxide, which is a method called FPOP, and Lisa Jones will be talking about that a little later on. Of course, you can do protease mapping or cross-linking, or HDX is another very common technique, similar to what we do. And then Jana Kiesler will be talking later on about this very new method of CF3 footprinting, which is quite interesting. So here at Berkeley Lab, we use X-rays, and we call our method XFMS, X-ray footprinting mass spectrometry. And I just wanted to note that footprinting has many forms, and it also has many acronyms. So it seems like no one's really decided on the common acronym for this method. So you'll see in the literature hydroxyl radical footprinting, hydroxyl radical protein footprinting, various other names. And one other thing to point out, so we look for modifications on residues. So you can see an OH addition of 16 Daltons, which is common on some of the residues, but other residues are modified in different ways. So you may not always be looking for a +16 addition. There's these subtractions and there's additions of other amounts. So just something to note for the data analysis later on. So the way we collect the data, we typically collect several different exposure times, and we monitor the increase in a modification as a function of X-ray exposure. Then we plot out the fraction of unmodified residue relative to the total. And what we get is this, what we call a dose response curve, and we're basically plotting the fraction of unmodified product as a function of exposure time. And so if there's a lot of modification of a particular residue, you'll see a steep curve to this dose response plot. If there's no modification, for instance, your fraction unmodified will be 100%, and you'll see a flat line. So typically what we do is we compare two different dose responses, or we compare two different states of the protein and look at the dose response of one state relative to the other. And that gives us information about where a ligand might be binding or where a protein might be interacting with another protein and occluding water at that point. So get a very brief example of how this can be useful in a particular system. So we've done a number of studies on this orange carotenoid protein, which is a pretty small protein. It's been crystallized in the inactive state, which I'm showing here, but not crystallized in its activated state. This is an example of some real data that we got when we looked at the active versus the inactive state of this protein. And you can see that if you look at the dose responses, some of the residues became more modified when the protein was photoactivated, and some residues became less modified. And so we plotted out that pattern of protections, and we were able to see that the pattern showed us that there was a lot more solvent accessibility in the activated state along the domain interface. So we were able to infer that the domains come apart as a function of photoactivation. And then if we look more closely at the residues that are lining that carotenoid pocket in the protein, and again, some residues became more protected and some became less protected as a function of activation of the protein. And the pattern was consistent with the domains coming apart and the carotenoid moving completely into one of the domains. And then this was later corroborated with the crystal structure of just that one domain. So this is trying to give you an example of how you can use solvent accessibility information to get structural information. Okay, so I think I stayed on time here. We've got a really great lineup of speakers. So I am going to hand it off to Cyan, and let me just introduce Cyan as well here. Except now, of course, I've lost my mouse again. Here we are. So Cyan, if you want to start screen sharing. So Cyan's going to give us a very technical talk on our, what we've been doing in developing our sample jet delivery method at the ALS. So Cyan has a bachelor's in chemistry, a master's in biophysics and molecular biology, and a PhD in biophysics and biochemistry. He was at the Center for Synchrotron Biosciences at the NSLS at Brookhaven Lab for many years, developing footprinting methods. And I feel very lucky that I was able to steal him away from Brookhaven Lab and bring him to the ALS, where he's been since 2013. All right, Cyan, take it away.
Thank you, Corie, for the great introduction. Good morning, everybody, for coming to this workshop. And today I'm going to talk about what are the improvements of sample exposure we did and why we did. So to start with, I just want to mention that this technique is a flux-driven process. So we need a certain amount of steady-state hydroxyl radical concentration, which is very important. So if you have more flux, high flux, short exposure, that will lead to low damage on the protein systems, and we get better data quality. So I want to focus on this one and how we can actually determine this flux issue when you come to do the experiment, and what improvement we need to do to get this high flux, short exposure, low radiation damage. The next, I want to point out that we started this technique, Jana and Mark Chance and I joined that time. We did actually start around 1999 with protein samples, and there we used to use its tube and X-ray, 1x1 exposure with an unfocused beam, low flux density beam. And then on 2006, we installed the mirror, and that gave us some high dose. So there are like 14-fold effective dose. And then from hundreds of milliseconds, we came down to like 10 or 20 or 30 millisecond exposure time. So that was a big improvement. On 2013, in Bartle Lab, we started working on the capillary flow, and we got further higher dose, like 30-fold effective dose, and we bring down the exposure time to microsecond. Now we are working on a new way of exposure, like a liquid jet, which I'll show later on. That is possible to get like even 1 microsecond exposure, and also there are a lot of other advantages. So Corie mentioned this slide. I want to point out what happens when you have too much exposure. So if you see this other reactions going on as secondary damage, like when this one recombines, makes hydrogen peroxide, also in the solvent electron that will consume oxygen, and this we need for protein modification. So too long exposure will lead to a lot of complication. You have secondary damage, you have loss of oxidation, so you have less yield of modification. So it's very important to get a short pulse with high concentration of radical. And so how we know that we have a good dose or not? So this was done in a very useful way using a fluorescence dye. So when you put the fluorescence dye in a different exposure time, the fluorescence of the Alexa dye or any fluorescence dye will be bleached. And if you plot the bleaching amount, and we found a red constant. So this red constant gives the idea that if sample is having enough dose or not. And we have been using this one in a very useful way just before the experiment. There are many experiments when user come, and we spend like 2 or 3 or even maybe half a day, because we have to tell the user that it's not good, and the user will go back and do the sample manipulation, came back to us. This is an example of how we can actually do the fine tuning using this Alexa dose assay. So here is a black curve. You see, user came with a protein in detergent, and we use unfocused beam, and we don't see any reds basically. So it's kind of a big amount of scavenging because of detergent. When he used the focus beam, we actually saw 2 different doses with a different protein and a 2 different environment, like protein in EDTA, we got 60, and protein with no EDTA with 144. So in this technique, we compare 2 samples, and it's very important to have a same amount of dose. So in this Alexa measurement at the beginning helped us to actually find out the difference in the dose. Like we can correct this factor that this manifold is difference in later data analysis part. So overall, it shows how the focus beam is important for the optimal dose and the correction that we need. But this whole process took like a lot of time. So just to give an idea about what kind of beamline configuration we need, it's pretty simple. We have the beam coming out from the storage ring, and we mainly use 1 mirror, focusing mirror, toroidal mirror, and that gives a focus at a certain distance on the sample at a red dot. And this was the configuration I showed here is X28C, the old NFLS beamline. And there we used to use this kind of multiple sample holder where you put tube 1 by 1, open the cap, and the bottom of the tube, you put the sample and you expose 1 by 1. And also now Eric will talk about more about this one. He has developed high throughput 96 well plate version of this. So you give the sample here in a well plate, and he does the exposure using a millisecond starter. And also we used to do in the capillary flow. So here, the same configuration, but you stack a capillary tube between a plate, metal plate, and cool down the metal plate so that the heating effect will not be much. So advantage of capillary is that, so this is shutter dependent. So you have a shutter dead time. So we used to go down to 10 milliseconds. But this is a flow dependent. You can move as fast as possible and then get a very short exposure time. So there's certain advantage of the capillary setup. So at the current, we are installing a similar focusing mirror at our ALS beamline, which will come up running at 2021. And we will be getting kind of pretty high dose, like power density will be around 30 watts per millimeter square, which is like kind of comparable to NSLS2, maybe a little bit less, but it's enough for doing microsecond exposure. So what we have been doing so far is that I told you that the capillary is stacked between metal plate. Here is the picture. You can see that we flow the solution and collect it, and there is a tiny black spot. You see there's a beam hitting the capillary. So sample is passing here, and all the temperature control is done by circulating water bath. So we got some usable minimum exposure of 200 milliseconds using this 200 micrometer inter-diameter capillary, glass capillary. So to give you an idea, what is going on with this capillary flow? So we need a flow. We are here because we cannot use too narrow capillary because this exposure time T, you can see inversely proportional to flow rate or the flow velocity. If we go too high flow rate, we have huge back pressure issue. Also, when you go for this capillary, there's a radiation damage. I can show you here in the next, in this video, you can see how the capillary will get burned. And in this picture, you see that there's a custom heat damage and a blackening of the surface. This is a problem. So because of this problem, you cannot actually go for a narrower ID and get this shorter exposure time, like 10 to 100 microseconds, even though we have the capability of the focus beam. So what we thought about, what happened if we remove the coating? This is a glass coating, which is like a huge compared to the inner diameter, and get a liquid jet. So we compare different capillary diameter, outer diameter, and try to figure out how much dose we can theoretically improve. So here's the plot showing that the flux density, without the glass, and this is the flux density transmission from the sample with the glass. So this is like 10-fold less what we get actually in the liquid jet. So we try to implement this technique and try to figure out what is going on. So another thing I told you that this capillary has an issue of the flow. So when we do the narrow capillary exposure, you can see that there is some non-linearity coming up at the initial exposure time. This is the fluorescence bleaching versus exposure time. So you drive the solution faster and faster, and you actually overexpose the sample. So what is happening? Some kind of solution is not flowing. So it's kind of a resistance coming over here. So this is like a Bunsen burner kind of stuff. You see that if the flow, when you light the burner, you say it's just laminar flow kind of a parallel, but sometimes it happens like cone. So what happens because of surface drag, resistance of the flow, it tends to slow down the flow. So you get more and more exposure. So this is very bad for our purpose because we need a linear dose. A linear because we need to compare different dose rate. We calculate the rate. So here I can show one example of what happened when you put this capillary in the beamline, high focus beamline. You can see the burning will be coming from the side. So you see that blackening out, starting from one surface and it's kind of covering other surface. This is the problem with the capillary flow. We cannot go for a very high dose. So after this burning is done, the sample will be charred or damaged and then clogging. So it's a huge problem with that. So then we try to find out what kind of containerless option we can do. So there are several options. We can actually do a liquid jet, just like a tap water. If you open the tap water, you see a big column of liquid. And then we can also use some kind of gas accelerated liquid. So you pass the gas. It's just like a mass spec ESI, but it's not a spray. You high pressure gas and you can pass a sample and the solution will come out from the nozzle. There are other ways we can do liquid sheet. You can also form a sheet to a capillary. It's like a very thin, tiny film. These are like continuous flow. There are other methods like inkjet, like a Dexter, like piezo droplet generator, so you can actually make different droplets, piezo generated drop, acoustic, like sound generated drop. So we tested this kind of stuff in our beamline, but the problem is that it's a discontinuous and the size is also small, and we need certain amount of sample for mass spec. So this is a 4 point that is best for XSMS. We need a tiny size is small. We need this precise speed control to get a dose response. We need a continuous flow. We need total correction time, not too much, because if I keep the beam on, then again, the sample damage will be coming back. So overall, when we did the survey, we found that Raleigh liquid jet is the best for our foot printing. So we have a size which is like 20 to 100, this should be micrometer. And then the speed, we can start from like 1 meter per second to 20, 50 meter per second, to a large variation of dose we can do. And we can collect the sample within 1 to 5 seconds. So this is like a sweet spot to do the experiment without, generating too much perturbation on the sample. So here is the jet that we do. The jet depends on if you can overcome the kinetic energy or the surface energy. So you're throwing a liquid from a nozzle, tiny nozzle. So if you have enough kinetic energy, it will come. So there's the upper limit of that, and also lower limit of that, and also upper limit. So like if you have too much force, then what happens? The shear between the air will actually make a spray. So this is the one, the red one is the upper limit and the lower limit. So we have to play in between this region. So I pointed out before that this particular region we couldn't do before, and now we can actually do it, the exposure from 10 to 100 microseconds using the liquid jet. So I want to point out that this particular region is very important. So this plot is in a log scale. So if you just do linear plot, you see there's a large variation we can do of the speed. So the dynamic range of the exposure time will be higher. So you can actually go for many fold exposure on the starting point. So you have better kinetics, so you have better quality data. So, but also we got to remember that when you go for narrower ID, you increase the back pressure. So here are some video of the jet we tried. You can see that if you have not enough speed, it's kind of a bubbles, the drops are falling. If you have also, it's not stable, but at a very high speed, you have a very nice jet, and it's staying for like very long, like 2 millimeter or more. I think short of time a bit. So these are the exposure of Alexa analysis we did when we have the capillary and we have the liquid jet. So we see that the black one, when you have the capillary, and the red one is a liquid jet, we see consistent increase of 2 to 5 fold of the dose with Alexa. So there is some increase. When you did the protein dose response, we saw variable increase starting from 2 to 10 fold. So it's kind of a pretty high increase of the dose in the protein sample. Also the quality. So we see that capillary flow, the amount of yield of the oxidation was very low compared to the jet. So it increases yield of the modification. And this is very important because when you do the derailment and mass spec, you need to identify those residues. And this will help our precise identification or sometimes more amount of oxidations. So we are going for the automation now. So why we need automation? Because as I told you before, this needs like 1 hour or maybe half a day to do the dose response. And our target for the automation is to lower the exposure time and lower the sample volume. We need a minimum amount of sample volume and a shorter exposure. With this in our mind, this is schematic. I'll not explain in details, but you can see we use mainly a light-induced fluorescence configuration of microfluidics. We excite the jet with excitation wavelength, and we collect the same light emitting from the jet to a fluorescence imaging device and use PMT and fast data collections. And this whole thing is controlled by LabVIEW software, and we can get a very fast automated dose response analysis. So here's some video of that. We are selecting the jet, we're getting the side view of the jet, and this is a microscope image of 50 microns. So this is the overall view of the configuration, which I will not go to the details. If somebody is interested, I can share the slide. And again, some nice, pretty jets here. You can see how long the jet is stable. And this is the experiment we did at NSLS2 beamline. The brightness of the flash density is so high, so it's ionizing the air. And you can see the jet is passing through that beam. So it's pretty amazing to look at that. Here, again. So in this, on the right-hand side, you see the current condition of the beamline. So there are a lot of wirings, but overall idea is that you're putting that jet, aligning it exactly the beam path and collecting automatically using a traction collector. Alignment is another issue, that this is jet is not as stable as a capillary because it can move micrometers. So we need to have a live alignment. So we are developing it using digital processing, so we can determine the center of the beam and also center of the jet, and then by manipulation of the software, we can actually align these two. And this is something maybe will be available next year to the user. I'll not go to that details, but this is the overall user interface that we made, very friendly. So you can just list the sample and just run it. It will do the exposure. And once it's complete, you can go inside a hut and collect the sample. So overall efficiency of the experiment became, like, I would say, 1,000-fold, because you don't need to go to the hut. You are getting everything on the bench. So here are different kind of a readout from the, the automated versus the manual. I just want to focus on the right side. You see that I don't see any difference. Kind of a similar response we are getting from the automated dose response versus the manual. So this is an example of the protein dose that we got when we went to NSLS2. So this is intact oxidation of cytochrome C. Cytochrome C is our standard sample. We got a huge amount of modification at 2 microseconds, and then massive with 10 microseconds. We are very happy to see that. So we try to see what happens site specific, and we tried different buffer like trees and herpes. These are like we don't consider trees and herpes because of their scavenging property. But at NSLS2 using the jet, we actually able to get a site specific dose of this residue specific dose, even at trees and in some sites and herpes. So this is a single digit. We can actually go for double digit. And with a double digit, we can get a better dose for the residues. Those that will show, dose in the herpes. Another thing, this needs more studies because we did only one protein. That we were able to detect more sites. So I think this is because of the high quality data. Because you have more oxidations, mass spec will detect more, sites which we are missing before because of low yield of the modifications. So we also tested the large complex like phycobilism, which is like antenna of the photosynthetic reaction center of cyanobacteria. And we are able to detect site-specific dose at different subunits, which is deeply buried inside the complex in the double-digit microsecond exposure time. So this kind of really promising results, and we need more samples to do that to try to see what's happening with this large complex as well as membrane proteins, which has detergents. So overall, we got like 10-fold increase in dose. We can use less sample. We can do automated tier. And overall, overall time of the experiment is now we can complete the experiment in an hour, using the dose response. That's pretty significant. There are different kind of future development that we want to do. We're going to have a mixing device that will collide to jet. And you can see this is with Dan DePonte. He's in Stanford Accelerator. He's developing this kind of mixing. We can use this kind of mixing jet to do fast kinetics. The other development we are doing is, so this is the current configuration. We are flowing the sample, we expose, we do a fluorescence of the Alexa after exposure. Another thing we are doing now, we are doing mixing. So we are mixing cell and we mix sample. And since these jets are very high speed, the mixing delay will be very less. This is very particularly useful for a pump and probe experiment where you can excite the molecule with the light. And then within a short delay, as 10 microsecond delay, we can actually do the X-ray probing by footprinting. And also, since the jet, there is no glass, you can do spectroscopy and do simultaneous spectroscopy and footprinting. Okay, so we are working with different group, different collaboration. Particularly, I want to thank Matthew and Brandon for it from Sonoma State University. So they worked on the automation and great achievement after they joined our lab. Corie, we had Arnold in our summer student here. And Chris is our mass spec collaboration at JB. Chris and Ian. Sean collected intact cytochromacy data. Daniel, I get help from this microcapillary microfluidic stuff. He's in our lab. And we went to NSLS2. I want to thank Mike, Eric, Donald, Auri for all their support for successful data collection. And we also work with Dan. I mentioned about this jet development. And Kevin in our advanced light source. Actually, we discussed this idea with Kevin when we wanted to start this process imaging. And all the users, actually, without them, we can actually test the sample. Thank you. I'm kind of hurry, and maybe I make it a little bit delay. Thanks.
Thank you, Cyan. That was terrific. So if you can stop your screen share. So we don't have a huge number of questions yet. So what I'm going to suggest is that we take a 4-minute break. Everybody needs break from Zoom meetings, and reconvene at 9:45 for Jana's talk, and then we'll field questions after that. Let's get started again. I am really pleased that Dr. Jana Kezelar can join us today to tell us about CF3 footprinting. So Jana is an assistant professor in the Department of Nutrition at Case Western Reserve University, and associate director of proteomics for Case Comprehensive Cancer. She's been pioneering hydroxyl radical footprinting methods for many years now, and most recently has developed hydroxyl radical footprinting methods that allow the study of historically challenging problems in structural biology, such as interactions of amyloidogenic proteins and intrinsically disordered proteins, which is quite interesting. And she is going to tell us today about another method she's pioneering, which is the CF3 footprinting. Okay, so take it away, Jana.
Hi, everyone. So I would like to share with you today a new technique which we developed in collaboration with Michael Gross, because he was a pioneer actually, but we are developing it and adapting it to synchrotron platform. So just kind of short outline, I'm gonna talk today a little bit about basics of trifluoromethyl radical chemistry, which we call CF3. For protein footprinting, then I'll talk a little bit about show you some experiment which we used to test the CF and develop this technique for synchrotron platform, and I'm going to show you data on a global intact protein, and also I'm going to show trifluoromethylation of the protein using bottom-up technique, which is a conventional for typical footprinting study, and also I will show you the comparison between CF3 and hydroxyradical labeling, and then you kind of have an idea when you can use one or the other chemistry. So this just slide again, just a kind of recap of Corie already said. So protein footprinting, general protein footprinting strategy is when you have some chemical reagent, and that chemical reagent in solution modify protein side chains accessible to solvent. Mostly on the surface of the protein. So then we digest, and those chemicals can be actually anything. It's not only hydroxyradical, you can use any chemical which can modify specific residues. So then we digest the protein. So, and you can see here we have in the peptides, we have some mixture of peptides with the various labeling within those peptides. So and then we use mass spec just to kind of collect our readouts. And we use tandem mass spectrometry to identify the site of labeling. So, and then we use MS1 to quantify the labeling. So we quantify it by extracting ions from chromatogram and for unmodified and modify, and then we do basically we calculate the fraction of unmodified or modify, and we plot this value versus the exposure time. So we call it dose response curve, right? And then we, as Corie already explained, so we compare those dose response curves for various conditions which we put protein in. So basically, as you can see, you can use any kind of labeling to do footprinting experiments. So here is a table from our recent review. We try to highlight main chemistries which is currently used for protein footprinting. So hydroxyradical is the main chemical reagent which currently utilized because again of the size, and it behaves similar to water molecule, and also it can be generated by various techniques. It's radiolysis, photolysis. You can use phantom chemistry. So you can use electrical discharge and label a wide range of residues. So basically you can label like 17 out of 20 residues, but typically we observe about 12 out of 20 routinely in mass spectrometry. So for various reasons, because some residues has very low reactivity and it's hard to label them. So maybe again, as Sayan said, the jet will help us to also label as very low reactive residues. So the mass shift of product we generate in this chemistry is quite complex for data analysis. So the second quite widely utilized now chemical is EDC and GE. So this EDC is a small cross-linker, zero length cross-linker, which typically used in a cross-linking experiment. So this chemical used to activate carboxyl group, make them nucleophilic, and then GE quenching this group, and then resulting in the production of plus 85 Dalton products, and also that's 85 can hydrolyze, and then we can sometimes see a plus 57 Dalton product. This is a little bit more simpler, I would say much more simpler than products we can observe. So it's mean that it's easy to detect it by mass spectrometry. So, but it labels only two groups, carboxyl groups, so as aspartic and glutamic acid. So we only can utilize this technique when we have proteins enriched with glutamic or aspartic acid. However, GE and EDC chemical you can control, so you can add different concentrations, so you can basically increase the labeling by controlling concentration, which is a contrast to hydroxyradical because you have a constant amount of hydroxyradical in sample. So when a protein too large, hydroxyradicals usually doesn't work well because you see only methionine, tryptophans, very highly reactive residues, and you don't see anything else, you lose resolution. So with G labeling, you can increase this resolution basically by adding this chemical in different concentration and labeling protein more efficiently. So, and there are other two techniques which are not as utilized, but still worth to mention. It's a methylene carbon chemistry because it's a residue, not all residues can be modified. So, and usually this reaction is done by photolysis of diazarine. So again, like some groups are using that as not as widely utilized as hydroxyradical GE currently. And carboxy chemistry only labels 6 residues, has only plus 72 product, so which is simple. But again, a reaction is kind of more complex than the described above reactions. So why I actually show you this list, because it is very important to understand protein footprinting is all about resolution. You have resolution, you have information. If you don't have resolution of your proteins, you don't have enough labeling, you basically lose all information and experiment become useless. So we're always looking to develop some chemistry to increase this resolution. So, and one of the chemistry which been actually pioneered by Michael Gross, but it's been done by other people before, but Michael Gross specifically pioneered it for proteins. So this chemistry utilizes trifluoromethyl groups, CF3. So those groups been used historically for just to study agriculturals or in pharmaceuticals, or design some specialty molecules, because when you incorporate this group into molecule, the molecule become more stable, changes the physical and chemical properties. So, and that's why this molecule was very kind of famous for using like in pharmaceuticals, for example. So it was a few attempts to try to apply this directly label the protein, and only Barron group was able to first show that this chemical actually can directly modify the protein, trifluorometallate the protein in a trace buffer and without changing functional activity of the protein. And that was the first work which opened up this field for protein footprinting. So Michael Gross, Davis group, and Kraska group, they all independently developed CF3 radical chemical to label proteins. So Michael tested this on using photolysis platform when he used peroxide exposed to the laser, generating hydroxy radicals. And these hydroxy radicals label sodium trifluoride, not like label, like make it radicals of sodium trifluoride. And this CF3 radicals react with the protein and labeling amino acid side chains. So also there is a secondary reaction going in this method, which it's a hydroxy radicals, which also generated in a sample. So hydroxy radicals react also with the protein the way we already know, and we also can observe some labeling with hydroxy radicals as well, and basically in parallel. So, however, since we add a lot of sodium trifluoride, so we really we add like somewhat from 300 to 3,000 fold excess compared to hydroxy radicals. So we're skewing the reaction into labeling protein with CF3. But you can see if we control this chemical concentration titrate, so we can actually change reaction, make it more like CF3 or make it more hydroxy radicals. So which is very important in this technique. So the next step was the problem with the photolysis platform. Everything is looks nice, but the problem is that we were able only to expose on a minute scale. So which it's not always compatible with the dynamic studies we would like to do. So we kind of thought with Michael, so what if we try a synchrotron platform and see if we can do it on microsecond level? And that's how our collaboration was born with Michael Gross. So we use, in synchrotron platform, so we use radiolysis to generate hydroxy radical. This is just the first step, which is different from photolysis. So as you can see here, the water molecule exposed to X-ray, and then we generate hydroxy radical, which undergo the same exactly reaction as the one I described during photolysis. But the only difference is in millisecond level right now. It's very fast reaction. So we decided to test our hypothesis that yes, we kind of thought that we probably can use synchrotron for and make it fast. But we decided to test it on a protein system. And we use some myoglobin in APA and HOA form. Again, this is just later, I'll explain why, because we want to kind of also correlate, make sure that we correlate this labeling into the structure. So, and the labeling reflects structural changes in the protein. So, and what we did, we expose this proteins in the presence of CF3, 20 millimolar of sodium trifluoride, and we exposed to 0 to 75 milliseconds. So, and then we did our typical footprinting experiments as we do for hydroxy radicals. So first of all, we also decide to take a look at the global scale. So what do we see in the labeling? So, and for this case, you can see we run myoglobin on a mass spec, intact protein. So, and I show here the spectra for the 20th charge state of upper myoglobin. The top panel show their upper myoglobin exposed for 0 milliseconds. You can see just the charge state with some maybe oxidation a little bit. So and then we have the middle panel shows the upper myoglobin exposed for 25 milliseconds at the beamline at Brookhaven. So, and we start to see some already incorporation of labeling. The number of CF3 modifications are indicated here by star. You can see we incorporated 1, 2, 3, 4, and maybe even more, but we can't really detect. So, and the bottom panel shows whole myoglobin exposed to 25 milliseconds. You also see that we observe incorporation of the product, and the product for CF3 is plus 68. So, it is obvious right away from this spectra that a wholer state of the protein is less susceptible to incorporation of CF3, which makes sense because a hollow state is more compact and an upper state is more unstructural. So that's why you can, even on the global state, you can evaluate overall labeling in a protein itself. And see which state is more which state is more compact or more open. So the next step we did, we follow our general approach Corie already described to you guys, so that we just digested all our samples with trypsin, and we run mass spectrometry in DDA full analysis mode. So, and here the slide shows just one example of extracted ion chromatogram for peptide 119 to 133 for the whole myoglobin. The spectra here, extracted ions are superimposed here. So we extracted actually ion for unmodified species for this peptide, and we extracted for only OH plus OH, and then we extracted also that peptide with the product of CF3, and then with a product which contained both labeled by OH and CF3. And as you can see here, methionine, in OH, we only observe methionine, and I'll talk a little bit about this more just in a minute. So then we also observe for CF3 chemistry, we observe phenylalanine and histidine, and they were predominant labels, and then we see some mixture of methionine oxidized and phenylalanine with CF3, and again oxidized histidine and phenylalanine with CF3. So you can see here that the pattern of OH and CF3 labeling can be quite different. So we see a different residues basically modified in this experiment. So this is the same exactly chromatogram, but now it's not superimposed, you just can see. So here is the methionine extracted for +16 Dalton shift. So, and you can see there is no any other labeling, because again, methionine typically gives all this bad tailing. So you cannot resolve any peaks for another residues, even if they exist there. But most of the time, methionine really mute labeling of other residues because it's so reactive. So when we extract CF3 +68 product, we see a nice clean spectra of various modifications. And also then now you can extract for a mix one, and also you nicely separate now the peaks, you can quantify them and you can have information with them. Why this is, I focus specifically on this peptide, because you can see, like, if let's say you have protein with a lot of methionines, and like, for example, some amyloid proteins like prions, they or like overwhelmed with methionines, or calmodulin, they have a lot of methionines, more than like an average protein. So in this kind of cases, most of the peptides would contain at least one methionine residue. So it's mean that you're gonna lose tremendously resolution in this protein. So in CF3 chemistry help to increase this resolution, because you're basically labeling completely different residues in this peptides, and you're muting methionine, because you can see methionine is not really labeled here. So this is the beauty of this additional chemistry, and it all comes basically in the same vial you exposed. So here's another example of extracted ion chromatogram for different peptide for whole myoglobin, and again we extracted for OH and CF3 products. It's a superimposed spectra here again. So you see unmodified peak, and you see all peaks which been labeled by OH and peaks which been labeled by CF3. You can see here the predominant peaks in OH is valine 64, and for CF3 is actually different residues, a histidine. And histidine and OH is very low here, like a blip, which is sometimes not easy to quantify. So, and also you have some residues which overlap. We see like leucine 72 and 76 labeled in both cases, right? So again, you have a pattern where you have some residue overlap, which is good because it gives you a reproducible replicate experiment in the two different chemistries, and also it gives you a different information. So, last example of another peptide. So this is peptide 80 to 96, very important peptide implicated from the crystal structure as a peptide which bind heme in hollow form. So, and this is extracted ion chromatogram for unmodified and then modified histidines. which again, very important. So, and also I show here dose response curve that you can see the difference in a whole versus upper form. We have less labeling with the CF3 in a whole form than the upper form. So we collect all this data, which we measure on a residue level, as you can see, not on a peptide level. Everything was done on a residue level. So we quantify modification rates for every single residue in whole and upper form, and then we took a ratio and we plotted this ratio versus the residues. Here you see some residues doesn't change, like insignificant change. Some residues show changes in whole form and labeling. Some residues show the decrease in whole form and labeling. So we compare this information to available crystal structure for myoglobin. So, and you can see here, so we depicted the residues here, and we color coded the labeling residues according to this fold change for labeling. So the blue indicate that we have less labeling in whole form, and red indicates that we have more labeling in whole form. So this important helix, it contains this histidines we label in this peptide, and this helix is actually important. It was implicated in the heme binding. So also there are some other peptides we look here, and the residues we quantify in the vicinity adjacent to this heme pocket. So they actually were showing some increase in labeling, so indicated some conformational rearrangement. So the same experiment we done exactly for upper myoglobin and whole myoglobin for hydroxy radical exposure only. So, and you can see here we actually went through the same protocols to collect our data. So, and we plotted again the fold change in labeling of whole versus upper myoglobin against the residues. So we also see some residues in a similar region which shows increase in labeling, and we see in a similar region decrease in labeling. But there were some differences also, because again the pattern of some residues were completely different between OH and CF3. So we have additional information. So we put this information on the structure again. In the same manner, so as I already described to you. So we see that some area adjacent to this heme binding so show also increase of labeling, but if you can notice that we don't see this helical at all labeled in hydroxy radical experiment, which actually implicated in binding to the heme. So if we only do hydroxy radical experiment, then we will lose this information completely. So we'd not even know, will not collect information for this region. So here, so I hope I convince you that actually, like we have in this chemistry, we have two different sets of labeling we observe. So for example, for hydroxy radical CF3, we have some residues which labeled with both chemistries, and also we have residues which were labeled uniquely, like 13 residues for hydroxy radical and 9 residues for CF3. So we saw total about 31 residues out of 153, which gives us about 20% of sequence coverage. But again, it's total sequence coverage. It's not what accessible to the labeling. So, and that's the number we usually try to increase to get more resolution. So if you only do, for example, hydroxy radical, you would miss all this part here. So, and if you do only CF3, so you would miss also like hydroxy radical labeling. So, but what is important to kind of show you, if you see here, there are two residues, histidine 119 and phenylalanine 123, which we label with CF3, but they're very reactive in hydroxy radical experiments. The reason we didn't see them because of this methionine, which is in the same peptide and basically muted our detection of those two histidines. With CF3 chemistry, we were able to nicely detect these residues and have information on this peptide, which otherwise would be lost completely to us. So, and also the other residues I shown here in red. So this is those three histidines, which located in important helix that binds heme. So, and it was completely absent in hydroxy radical experiment. We just didn't label as histidines very well, or we cannot detect them. So this is just kind of to show you how important to have actually a parallel chemistry, and then also CF3 labeling becoming like really essential tool if you guys have a proteins with a highly enriched with methionine residues. CF3 experiments become the experiment which solve your problem. And we actually have a lot of difficulties with the prions recently, because for the same exactly reason, prions has a lot of methionines, and that's all information we can have. And CF3 shows that we can have multiple tyrosines. We dramatically increase the coverage of prions doing this chemistry. So the second question we will try to answer here is, do we have a correlation of CF3 labeling with the structure? Because this is very important. We all know that hydroxy radicals labeling correlate very well with the solvent accessibility. The question here is that, does CF3 chemistry correlate with solvent accessibility? First of all, we saw all the residues we identify label, we found to be solvent accessible from the crystal structure. So the second, as you can see here, the very important region which shows either binding interaction with the heme, and this region showed the change, decrease in a labeling in a holo form, which correspond to the binding of the heme and closing of this helix, and also like two adjacent regions to this pocket, which shows rearrangement of the protein in holo form, and then in the crystal structure they were showing to have more solvent accessibility. So we see the increase in labeling with the CF3 in the upper myoglobin compared to the whole myoglobin. So we established a very, very nice correlation, but using just expectant whole and upper forms, we established great correlation with also structural information when we use CF3 labeling. So in the conclusion, so then we have a little bit time for questions. So overall, CF3 labeling we found to be as a really true complement to hydroxyl labeling technique. So again, I want to again highlight that we see a pattern of labeling with the CF3, and we have pattern of labeling with hydroxy radical in the same reaction. So which kind of open up you to test a completely different regions on the protein and increase resolution. So also CF3 modify, for example, normally inert alanine in our studies here, but we also on other proteins and peptide studies recently we've done with CF3 labeling, we found that we also see some serine labeling, glycine labeling, which you typically don't even observe in hydroxy radical experiments, which again open up us to a different level of resolution. So hydroxy radicals also extremely reactive with methionines and often mutant the reactivity of residue proximal to methionines. By trifluorometylation, like modify methionine really minimally and allowing residues near the methionine to be observed and analyzed. So, and also CF3 labeling can be tuned by adjusting sodium trifluonate concentration, the same as GE and EDC chemistry, if you remember. So it's mean that, for example, when we study a large protein complex, like recently we're dealing with a chromatin remodeling protein complexes, which is like a mega Dalton system. So if we try to hydroxy radical footprinting, so, and we found that we typically modify only highly reactive residues. We completely lost resolution of the residues. So GE labeling was a way to go for us, and now we're gonna test the CF3 labeling because we believe that we can improve again coverage now for these large complexes by doing CF3, because again we can adjust concentration. It's just a chemical, you can change concentration and solution easily. So and lastly, CF3 footprinting experiment, I'm sorry, I just completely, so nicely correlate with the structure. So we're still trying to assess if we can actually quantify solvent accessibility like we do with hydroxy radicals. So we're not at the stage yet because we're trying to determine reactivity. And I can tell you right now, we see the reactivity of tryptophan is the most reactive, histidine next reactivity. Then we have phenylalanine, tyrosine label, serines. So we're still working now to establish this ladder of reactivity, which we're going to publish soon. So, but overall, this seems like this technique is very, very complementary and reflected the changes in solvent accessibility. So at the end, I would like to acknowledge people who were involved in this project. We exposed the first protein and the study we published. So we exposed an ALS, and Sayan and Corie help us. And current study we're doing at BNL on the peptide and protein labeling with CF3. So we also, Michael and Min Chan gave us great support and collaboration, and we published paper together doing the study with CF3. So, and also from CASE, I would like to acknowledge Mark and Aubrey, the people who gives intellectual contribution and did some studies, Aubrey exposed sample and did some analysis on this protein system. So, and I also happy to take your questions, and thank you for your time today.
Great, thank you so much, Jana. That was really, really interesting. So we are running late. I was actually going to suggest that we go right into Lisa's talk, and I see there's a couple questions in the chat window, but if people can just hang on for another talk, then maybe we'll catch up and we can answer all these questions at the same time. So Lisa, if you can go ahead and share your screen, and I will introduce you. So I'm really, really pleased that Dr. Lisa Jones can be with us today to tell us about some in vivo footprinting work. So Dr. Jones is an associate professor in the Department of Pharmaceutical Sciences at the University of Maryland. And her research focuses on developing in vivo protein footprinting technology to study disease pathogenesis. All right, take it away, Lisa.
Okay, can you hear me?
Yeah, I can hear you.
Okay, so thank you, Corie, for the introduction and the invitation to speak here today. So I'm very happy to talk to you about some in vivo footprinting work that we do in my lab. So we do, let's see if I can get my slides to move. We do use hydroxyl radical protein footprinting. And so I'm not going to go into it in too much detail, but here hydroxyl radicals modify the side chains of amino acids. And so you do end up with an oxygen on your side chain. And when you do mass spec analysis, so you will see modifications of +16, although that's not all you will see. There are multiple ways to generate hydroxyl radicals. In my lab, we use fast photochemical oxidation of proteins, or FPOP. So from here on out, I will call it FPOP. And so FPOP was developed in the lab of Michael Gross at Washington University in St. Louis. And what happens here is we take H2O2, hydrogen peroxide, we add it to our protein sample. We flow it through a flow tube, and then we irradiate it with an excimer laser at 248 nanometers. And so that splits the peroxide and forms the hydroxyl radicals that'll now modify the proteins. And so we do want to study proteins in their native conformation, so we don't want to unfold them. So we have several experimental controls to limit overoxidation. And so one of those is to add glutamine as a scavenger, and this controls the radical lifetime. So based on the reactivity of glutamine with hydroxyl radicals, the decay is at the 1 microsecond time scale. So we are labeling things very fast, faster than proteins can unfold. And so in that way, we can study weak interactions with fast off rates. So just to give you an idea of how FPOP works, so generally we do use 50 microliter volumes. We had buffer. There are a lot of buffers that will work. There are some that won't because they will scavenge the radical. We mainly use phosphate buffer, and we have our scavenger. So just prior to the reaction, we add our hydrogen peroxide, put it in a syringe, and begin to flow it through a flow tube. So we do use a pulsed excimer laser. and the system is under constant flow. And so that is another experimental control to reduce over-oxidation, because a bolus sample only sees laser radiation one time, so it doesn't get over-oxidized. You'll also notice this exclusion fraction here, which never gets irradiated by the laser. And this is wide enough so that radicals from this part of the sample cannot diffuse into this part of the sample in the time frame of the experiment. And that is a third experimental control that we have to avoid over-oxidation. Once the reaction is done, the sample is collected in tubes that contain catalase, which quench excess hydrogen peroxide, and methionine that quenches excess OH radicals. And so at this point, we do want to localize where the modifications are. So we do digestion with trypsin. Sometimes we use lyc coupled to trypsin into peptides, and then we do our LC-MS analysis. We tend to do, for our chromatography, reverse phase, which you see helps us when we see our modifications. So in this peptide, you have this phenylalanine modified, and you will see a shift in the chromatogram. And so this helps us distinguish FPOP-based modifications versus in-source modifications that the mass spec itself can put on, specifically methionines. So it's nice to see this shift in the chromatogram. And then we can do our quantitation based on the area under the peak here. And then of course we use our MS spectra to identify what's been modified. So we can get down to residue level quantitation. And so the type of information you can get here, FPOP can be used to identify protein-ligand interaction sites, protein-protein interaction sites, and regions of conformational change. And so you do want to do the differential experiment where you're comparing. In this case, you need to do FPOP on the protein by itself and the protein in its dimer form, and compare the labeling pattern between the two, because you would see a decrease in labeling at the interaction site because of the decreased solvent accessibility. So to date, there are a handful of groups that do FPOP, and they're doing them on purified proteins. So my group is interested in extending this into the native cellular environment so that we can understand how the molecular crowding in the cell influences protein interactions. And so this requires getting hydrogen peroxide inside the cell, our laser light inside the cell, so that we can generate our hydroxyl radicals in situ. And so there were a lot of modifications that we had to do to get this to work, actually. I'll just talk about one that we did, where we needed to develop a single cell flow system. So we still wanted to flow our cells past the laser aperture, but this is difficult with some cell types that like to clump together, and so they wouldn't have equal exposure to laser radiation. So we did this based on hydrodynamic focusing, where you have a sheath buffer that flows on both sides of your cell stream, and it flows at a faster flow rate than your cells, and eventually it'll drag your cells along one at a time. So using this, we designed and built a flow system that was compatible with our laser. And as you can see, it actually has two stages. So in the first stage, the cells and H2O2 mix in this mixing tea. This was important because our cells have endogenous catalase. So if we put the cells and the H2O2 together in a syringe, by the time they reach the laser, all of the H2O2 would have been broken down, or the majority of it. So in this way, with the based on our flow rate and the inner diameter of this tubing, cells and H2O2 are only in contact for 1.7 seconds prior to laser radiation. So that's not enough time to break down the peroxide. And so then when it comes into this second stage, into this cross, this is where it sees the sheath buffer. And so as it exits here, the cells should be flowing in a single file. So to test this, we did fluorescence imaging, where we added a green fluorophore to our sheath buffer and a red fluorophore to our cells. And as you can see, our cells are flowing one at a time. So now when we do what we call in-cell FPOP or IC FPOP using this single cell flow system, we get hundreds of proteins modified. So this is on Vero cells, which are kidney cells from the African green monkey. So this is our workflow here. So I just talked about, starting with our cells and our flow system. So what we do after we collect, here we use a quench that are cell permeable quench agents. And we do cell lysis, and then we purify out our protein fraction from the cell. and we digest the entire protein fraction and run the entire lysate on the mass spec. And we have to do this because there are no good enrichment strategies for oxidatively modified peptides. And so that does make our sample very complex. So even though we've had success in seeing how many proteins are modified, we are still probably missing things because of the complexity of our sample. And so what we found is if we actually do an offline fractionation using high pH reverse phase, and then an online low pH reverse phase coupled to the mass spec, we can actually pick up more modifications. So these are two different breast cancer cell lines, and you see we're actually modifying thousands of proteins within the cell. And so this is very good, so we can get structural information on many proteins in the cellular environment. We also wanted to determine that we were labeling proteins in the different organelles. So we didn't just want to be labeling extracellular proteins or proteins in the cytoplasm. If there was a nuclear protein we wanted to study, we wanted to be able to modify it. So in order to test this, what we did is we did organelle fractionation. So we did our FPOP on our cells, and then afterwards we did use density centrifugation in order to separate the organelles into multiple fractions. And then each one of these fractions was purified out and analyzed on the mass spectrometer. And so what we found across 2 biological replicates is that we actually get hundreds of proteins modified in each fraction. So we are modifying proteins in various organelles. So now we feel like in-cell FPOP is a general strategy that can be used to study a wide variety of proteins, no matter where they're located inside the cell. And this, coupled with the fact that we can get structural information on thousands of proteins in a single experiment, we think it'll be a good tool for proteome-wide structural biology and looking at things on a more global systems biology kind of a scale. And so one application we think this will be useful for is drug target engagement, where you add your drug and just see all the structural changes that happen in response. And so I'm just going to quickly go through this as an application. I'm not going to go into detail, just to show you the power of the method. And so here we're looking at Gleevec, which is a cancer drug, who it was designed to inhibit the kinase activity of BCR-ABL, although it turns out to be very promiscuous and does bind to a lot of other things. And so here we tested them on breast cancer cell lines. So this is triple negative breast cancer. That's what TNBC stands for. And we did 2 different cell lines. The EA is derived from a Caucasian patient, and the AA is derived from a Black patient. And we wanted to compare the 2 cell lines because preliminary studies have shown that Gleevec does not work very well in African-American patients. And so we did the differential experiment. In each cell line, we did in-cell FPOP with and without drug, and then did a comparison. So first we looked at the global oxidation changes with this volcano plot. So this is just a fast way to see everything that's changing. You see some proteins had lower oxidation after Gleevec treatment, some had higher oxidation, so they are changing in response to the drug, and we see that in both cell lines. The ones that are highlighted are interesting because they have opposite effects in the cell lines. So for example, if we highlight pyruvate kinase here, you see in the sample from Caucasian patient, it has a decrease in oxidation, but in the Black patient, there's an increase in oxidation after drug treatment. So this is evidence showing that the drug is working differently between the 2 cell lines. And so, but one of the advantages of FPOP over other proteome-wide methods is you can go from this global level data and actually localize on the protein where these structural changes are occurring. And so this is what we did here with pyruvate kinase, where we just did peptide level quantitation, and we can see where these changes are actually occurring. So the most important region is this peptide 476 to 489, because this is where the allosteric activator of pyruvate kinase binds. And so the binding of this activator is actually regulated by phosphorylation. So you would expect to see differences if you treat with a kinase inhibitor, right? And this is what you see here in the Caucasian patient cell line. In the absence of Gleevec, This allosteric activator is not bound, and this residue is actually the tryptophan 42 that specifically gets modified. So this is solvent accessible, and you get a good amount of labeling. Then when you add Gleevec and the phosphorylation pattern changes, now that makes way for this allosteric activator to bind, and it's no longer solvent accessible. What's interesting is you do not see this effect in the Black patient. Actually, it is highly solvent accessible, so this allosteric activator never binds in the pocket. So that means there was never no phosphorylation change in the cell after treatment with Gleevec. And so this shows that Gleevec is just not working in this cell line. And so we were doing expansion studies on more cell lines on actual patient samples from our medical school to see if this is a something that happens across these two racial populations, and try to understand why Gleevec does not seem to be working in the Black population. I also want to point out we have in the same data set been able to look at the phosphorylation pattern changes and have gotten information there. And that just demonstrates what an information-rich data set this is. You can get the structural information as well as the expression abundant differences and the PTM changes. And so that just shows the potential power of in-cell FPOP for proteome-wide studies. So we also wanted to see if we can go from in cells to actually in vivo in an animal model for human disease. And for this, we chose C. elegans, which are members of the nematode family. And they are used for a model system for many different human diseases, including cancer, Parkinson's, and Alzheimer's. And so this is a transgenic worm that expresses GFP in its neurons. And so as you can see, the worm is transparent. So because of that, we can get our laser light inside the worm. So the worm will uptake H2O2 through its skin. It will also ingest it. So that means we can generate hydroxyl radicals inside the animal and be able to do structural studies. So we did have to, of course, work on the flow system here, which my grad student worked really hard on to get the worms to flow. When you're dealing with the live animal, this was difficult. And this was even an initial one where we got clumping. She was able to remove this clumping, and we get one worm flowing at a time. We had to optimize our laser pulse conditions, and we found that a 50 hertz pulse worked the best, and we can get a little over 500 proteins modified in what we now are calling in vivo FPOP and IV FPOP for short. So this was great to see. So we can actually do FPOP in an animal model for human disease and really study disease pathogenesis with this method. The thing about these proteins is they actually come from all the body systems within the worm. And so we can study a wide variety of things. You see most of the proteins are in the digestive system, which we would expect since the worms are actually eating the H2O2 and taking it in that way. But you see a lot are also in the neurons, and this is accounted for the fact that the H2O2 is also absorbed through the skin. We did recently publish a paper where we added penetration enhancers to increase absorption of H2O2 through the skin, and we actually increased the number of proteins modified. I can't remember by how much. So this workflow where we have our flow system has worked well for us with the cells as well as with the worms, but it is very slow. It does take 10 minutes a sample. And so if you want to study fast biological processes like signaling and protein folding, which happens on the millisecond to second time scale, that 10 minutes just doesn't help you. So we wanted to develop a new platform for in-cell FPOP that allows us to study label protein samples much faster, or cellular samples faster. So we went to a static platform using one of these stage top incubators that we got from Oko Lab. So these are traditionally used for fluorescence imaging, and they allow you to do cell culture right at the microscope. So we custom designed this with Oko Lab, and they fabricated it for us for one that's compatible with our laser. So basically, the biggest thing that was changed was the glass top was changed out for quartz. And so it does hold 6-well plates. So now we can do cell culture right at our laser platform. So it makes everything much faster. This is a completely enclosed system. There's temperature control, humidity control. That's what this big line is. We can also pump in CO2 so that our cells are happy. And then you see we have these lines where we can now input our H2O2, our quench, things that are needed for FPOP. and any treatment if we want to do drug treatment in this case. So this is just what the whole platform looks like. This is our laser here. You see our stage shock incubator. So it actually sits on a movable stage, right? So because we have a fixed irradiation, but we want to irradiate each well individually. In that case, we can do time points as well. And so then this stage moves each well individually into the pathway of laser irradiation. These are our peristaltic pumps, so we can input H2O2. We have our CO2 tank, our temperature control, and humidity control. So we actually call this platform PIXI, for Platform Incubator with XY movement. And so really, so this has been very nice. And what we found with PIXI is we can actually label each sample in 20 seconds. So we've gone from the flow system from 10 minutes down to 20 seconds. We did want to see if we can get that faster, especially to look at protein folding over time in a cell. So we did automate this system. So I'm going to go ahead and show a movie. So what you'll see here is the different color solutions flowing in. Should be a laser flash, which you didn't see, but then there's movement of the plate to the next well. Maybe this round you'll see a laser flash. No, not really, but trust me, there's a laser flashing. So we've now automated this platform, and another thing that we're hoping is that that automated platform will make the method easier for other labs, so it can be translated to other labs because now they have an automated platform. So with FPOP, and we do in-cell FPOP and PIXI, we actually get more proteins modified. We get a 1.4-fold increase. We've also done this with the worms, and in the worms, we get a 1.5-fold increase in the number of proteins modified. So we do have FPOP working in this system pretty well. There are still some other parts of the automation where we're trying to get working, but we are very happy with this system, and it'll be very useful for studying signaling and protein folding in the cell. So in summary, IC-FPOP can be used as a tool for proteome-wide structural biology. We've shown that we can detect structural differences in the cell upon drug treatment. We can also do this in C. elegans for in vivo FPOP, and we feel like this new PIXI platform will give us a lot of flexibility with our biological applications, but also may be translatable into other labs so they can do this method as well. So in way of acknowledgements, I'd like to thank my group. So Emily had worked on the triple negative breast cancer cell lines. Dante really assembled and designed PIXI and got it automated. And Jessica, who's done all of the in vivo FPOP work. And our collaborators, as well as the funding. And thank you for listening. I'll be happy to answer any questions.
Wow, thank you so much, Lisa. That was super interesting. I had no idea how far you've taken the that in vivo footprinting. Wow. So I know you have a time constraint today, so let's ask questions of Lisa first before we scroll back to look at the previous questions on the previous talks. I have one to start out, which is, what happened? Is there a problem with lack of oxygen in cells? My understanding was that you need oxygen for some of those hydroxyl radical modifications to happen. And I know there's various levels of oxygen in various types of cells, but I don't know if you could speak to that just a little bit.
Yes, that was the first thing that we had to deal with. That's a great question. That was the first thing that we had to deal with. And I'm going to come back here because, so the thing about FPOP is that how catalase breaks down H2O2, it breaks it down to water and oxygen. So by doing this with FPOP specifically, you're generating the oxygen you need to do the experiment. Okay, so that's why it works with FPOP, because you are generating oxygen. So when we designed this flow system and decided which time, worked on how much time cells and H2O2 were going to be mixed, we had to balance it out, because we do want some of the H2O2 broken down so we can generate oxygen so FPOP will work, but we still need enough left that we can modify a lot of proteins in the cell. So in designing this, that was the thing we had to work out.
Okay, thank you. So that's really interesting that the FPOP will work for the in vivo, and I wonder if the X-ray footprinting will not work so well for the in vivo type of studies.
I think I think that's possible. It will be more difficult to do it with the X-ray because of the lack of oxygen.
Okay.
Yes.
Great. So let's see. Do we have any other questions for Lisa? I don't see specific questions for her in the chat window. Let's give everybody a chance in case anyone wants to unmute themselves and brave a question. And also, of course, feel free to continue to type in the chat window.
Can I ask a question?
Yeah, go ahead.
Like with heme proteins, I know they're really reactive with H2O2. Have there been any sort of different results with heme proteins, or they work just fine as well?
They do not work just fine. So what was interesting, the first heme protein we tried was cytochrome C. And so we got very interesting results with that because we, when you add the H2O2 itself, it changes the structure of cytochrome C because it interacts with the heme. But what we found is cytochrome C, has peroxidase activity, and that's kind of a hot subject right now that people are working on. And when you add H2O2, it induces the structure that it needs for its peroxidase activity. So we just published a paper in Biophysical Journal where we coupled in vitro FPOP with ion mobility for cytochrome C, and we were able to separate out the conformers after FPOP. So we actually can see the peroxidase active conformer from just adding H2O2 versus the regular function of cytochrome C. So heme proteins are very difficult to work with, yes, because of that, the reaction of the heme with just H2O2 by itself.
Which goes to show there's advantages and disadvantages to every single experimental technique.
Yeah.
So we have one more question in the chat window for Lisa, which is, what are the cell number requirements for the in-cell work? Okay,
so right now we're using 1 million cells, which seems high. I'm not sure if you think about it as a someone who does cell culture or not. So actually 1 million cells is a confluency of a single layer on this well on a 6-well plate. So if you're doing cell culture, that's not a whole lot, but it is a lot, 1 million cells. And so in the next step, now that we're open from COVID-19, that's what we're working on. How low can we go? Obviously, mass spec itself is moving towards single cell proteomics. Can we do that with FPOP? And so that's something we're moving towards. Just because of the variability between cells, you want to see if you can see how low you can go. But right now, it's 1 million a sample.
Great, thank you. So I guess in the interest of time, we should, I'm going to go to the other chat questions for the other speakers, but I want to let people know I have my email address in the chat window. So if you have more questions for any of the speakers that don't get answered today, please feel free to email me and I'll pass along your contact information to the speaker so you can talk to them directly after the workshop. Okay, so just real quickly here, a couple questions about CF3 for Jonna. So let's see here. We have, can we conclude the CF3 reagent acts as a radical scavenger, a radical quencher? So in that case, do we not need to use to do our usual quenching after the experiment?
Yes, CF3 actually reagent act as a scavenger, and we did some studies with a different attenuation, and we found, and then we look at the oxidative labeling versus CF3 labeling. So the attenuation, we have to really low attenuation for CF3 dramatically. So at least, like let's say we use attenuation of aluminum for attenuations, so about 500 microns. So here we use about 76 micron. You can see the range of attenuation. So we have to really open up the beam and make sure that we have the hottest beam as possible. So yes, but we're still quenching after we done chemistry, we still quenching with methionine amide, and because again you still observe oxidation of methionine with a hydroxy radical. So, and just to kind of prevent methionine from further oxidation in solution, so that's where we add quencher. But otherwise, yeah, we will consider in the same thing, just not to add quencher, and see how it goes. And we're testing this on a peptide level right now and see if we need any additional quencher for this. Okay. Nice question.
Yeah, great question. Thanks, Eric. So there's another question from Cheyenne, but I'm gonna actually hold off on that. I think we need to move along to the next set of talks, and I promise after this next set we'll take an actual break so people can get a break from Zoom. So what I want to do now, I'm going to share one slide. So I was going to tell you guys how to access the ALS beamline. And instead of going through a presentation, I'm just going to share one slide. So I just wanted to let you know that we're basically open for business at the ALS. If you want to learn more about what we're doing, you can, of course, email us to check out our webpage, alsfootprint.snappages.site, and you have our contact information there. To do a footprinting experiment at Berkeley Lab, the best thing to do is to go through the Molecular Foundry website. So if you go to foundry.lbl.gov, you'll see that you can actually go to their user program and read all about user guide, how to submit a proposal. And those proposals, you can send in a proposal for X-ray footprinting, and basically we have an agreement with the ALS, between the Foundry and the ALS. So you don't need to send in two separate proposals. You can just send in one, and we'll work with you on trying to get you up and going with a footprinting experiment. And I just wanted to point out real quickly here our team at Berkeley Lab. So Cyan, who you've heard from, Matt and Brandon, who did a lot of instrumentation work, Lena Christensen, who you'll hear from a little later on with the data analysis. And we have our mass spec expert, Michael Connolly, here at the Foundry, and our protein engineering expert, Behzad Rad, here at the Foundry also. I just feel really privileged to work with such a great group of people here. So that's all I'm gonna say, because we're running out of time. I'm gonna hand it over to Eric now to talk about accessing the footprinting beamline on the East Coast at Brookhaven Lab. So Eric, if you wanna go ahead and share your screen. And now we're back on time.
Okay. All right, so I'm going to say a little bit about X-ray footprinting on the East Coast. So Corie gave me 15 minutes, so I'll tell you a little bit about the beamline and how it differs from the ALS resource, and then say a little bit about access to NSLS2. So first off, I'm going to start with the acknowledgement. So we have acknowledged members of the CSB XFP team led by Mark Chance at Case Western Reserve University. Here at NSLS2, besides myself, we have Mike Sullivan and Don Abel. Today is Don's last day on the beamline. He's moving to a new position at NSLS2 starting Monday. And we have Rohit Jain, who's a postdoc focusing on science applications of footprinting. He's based at Case Western, but he visits Brookhaven a lot. And in the past, Jen Bowen contributed a lot to the development of the beamline, as did Sean Toomey. So at NSLS2, we wouldn't have a beamline without a really outstanding construction project team back in 2015 and 2016. And I also want to acknowledge Sean McSweeney, who's leads the structural biology program at NSLS2 for his support and interest in the program. And just as a note, funding, the beamline was constructed with an NSF major research instrumentation award, as well as fund from the state of Ohio, and operation both from the NIH and more recently from the Air Force Research Lab. So what you see here is an overview of NSLS2. And the little circle that I have there shows the location of the structural biology beamlines, in particular XFP. So this is a billion-dollar machine completed in 2014, been operational since 2015. About 28 beamlines operational right now, and about half built out. With scope for about 25 to 30 more. So one of the things I want to point out is this kind of was a greenfield facility, and when we were laying out locations of beam lines and so on, one of the things we wanted to do was co-locate similar beam lines in a similar area and kind of copy something that was done at Diamond in the UK and create these villages, if you will. So we have what we used to call the biology village, and they're all clustered right here in 745. You have a set of facility beam lines under Center for Biomolecular Structure that's led by Sean. So you have 2 protein crystallography beam lines and a solution scattering beam line. You have XFP, which is a partner beam line operated by Case Western. And you also have a 3rd crystallography beamline, NYX, that's operated by the New York Structural Biology Consortium, led by Wayne Hendrickson, right up here. And then we have also some useful support facilities. So there's new wet labs that were completed last year, right in building 745 in the green box. And just earlier this week, opened up to accept user proposals, a new New York State DOE BER funded cryo-EM facility in a new building located right outside 745 and connected to the building. So one of the things that this co-localization allows, I think, is, you know, what, and I'm going to say a little bit more about this later, is, you know, easy use of multiple techniques to answer questions. And you'll have somebody come in, do crystallography or solution scattering, and they will hear about what footprinting can do, and it offers an opportunity for them to, you know, easily get in touch with us and start doing it. So this here is just kind of an overview of the beam line. I'm not going to go through it in gruesome detail, but the key points is that we use a 3-pole wiggler source. So this has a critical energy of around 6.8 keV. So we have a usable flux curve that you see in the upper right here, photon about 4 to 4.5 keV up to 16 keV, and that's defined by the mirror cutoff. So that's a pretty much constant thing. And the mirror is located in the front end. We can bend it to provide vertical focusing depending on the experiment. Then we have 2 distinct end station. One we call the high dose end station, and one we call high throughput. And these differ in the amount of flux density, basically controlled by both vertical size, which we do control, and the horizontal size, which we don't. The horizontal size is simply governed by the distance from the mirror in the experimental hutch. So continue on. So this is just an overview on the left side of the XFP experimental hutch as it exists today. Corie complimented me on how neat it is. So we have the high dose footprinting end station is further upstream, and the premise here, so I kind of went through a lot of this, so I'm not going to go through it in great detail, but this sort of, this is the area where we've hosted his jet experiment, where we do from time to time capillary flow. The premise is essentially that the sample flows through focus beam of a defined cross section, and the velocity defines the exposure time. So I'm not going to go through specifics here, but you know, the key thing, and one of the things that, you know, I think makes the jet approach very compelling is that at NSLS2 and XFP right now, we can deliver up to 400 watts per square millimeter of power to the sample. Now, leaving aside what that may or may not do to your sample, let me just say that glass capillaries do not hold up very well to that at all. So, and that makes doing experiments at this end station quite difficult because we have very rapid capillary damage. And I just show a little schematic here. It's just showing the typical arrangement with a syringe pump and a capillary flow cell and a little sample collection. And we can also support some BSL-1 level in vivo flow experiment. This is mainly developed and used by Sarah Woodson for nucleic acid footprinting and ribosome assembly. So now I'm going to talk a bit about what is our pride and joy on the beamline, and this is the high throughput footprinting end station. This is what pays the bills. So the premise here, and Cyan mentioned this a little bit earlier, that we have a fast shutter that provides millisecond exposure of samples in the steady state. So, we can use it for a variety of things, somewhat less scavenging soluble proteins, studies of drug target interaction, freeze quench. One unique thing that we support at XFP is samples requiring BSL-2 conditions. So if you're studying prions or certain infectious viruses, that's something we can do. But the premise here is that the sample resides in a 200 microliter PCR tube. There's a 5 microliter droplet on the bottom, and the diameter of that droplet is about 2.5 millimeters. So we match the beam size, which you can see is this heart-shaped feature in the middle of the screen, and center it on the sample. So the droplet sees roughly even intense amount of beam, and also the sample depth in the PCR tubes is well matched to our 4 to 16 keV energy range in terms of penetration, depth, and absorption. So the device itself is in a 96 well format, so we can take plates, strips, tubes, and we can cool it, so it can go as low as minus 35 C. Maintain it at room temperature. We can maintain it at 4 C. And we also have it in a little chamber here that we can flow inert gas to reduce humidity and condensation. So I'm going to show a little movie here, which may or may not work. This just shows it in action. So what happens is it basically moves into each position, and then the fast shutter fires, and that's a view of the actual ionized pink beam and, at each shot. And these are relatively quick, so they may or may not come through. And so we can do an entire plate in about 5.5 to 6 minutes, depending on conditions. So one of the things we've developed over the past year I want to emphasize is we've spent a lot of time developing supporting infrastructure, that was something that we call BIFs, beam intensity filtering, shuttering, and slits. And the idea here is, the fear that we always have is these uniplex fast shutters that we use don't necessarily have a lot of external feedback. So did the shutter open? Did it open for the amount of time that it was supposed to open? Did we put beam on the sample? So that's the whole idea of this whole device here. We have, from the left side of the screen, we have an automated attenuator wheel that allows us to externally select up to 8 different attenuations without manual intervention. You don't have to go into the hutch. We have a diamond viewer assembly right after that, so we can view beam as it comes out the exit window. We have the uniplex shutter, and then we have a couple little tools that allow us to monitor whether or not the shutter opened after it, and we can also set up slits that allow us to adjust beam size or cut the trim beam size if we want. And then this proceeds down a helium flight path to interact with the sample. And then this just shows a quick overview of the interface that we have for this device. The thing with 96 samples is sample management, and setting up with the experiment becomes a challenge. Each sample is different in each cell. The exposure time is different. The attenuation may be different. We have this relatively simple GUI application right here that allows you to individually select and define conditions for each position on the plate, or you can simply assemble an Excel file and import it, and it will automatically select and the positions you want to expose based on your information. You can also export metadata, such as notes about the sample, the exposure time, a variety of other things. The other thing that we've done is a very simple one-button alignment that the user can come in, and after we've aligned the beam line and they're working on the experiment on their own and we're not around, I simply can press this align button and it will automatically move to this particular hole on the array and measure a beam profile through a little 1 millimeter hole that we have cut in it, and find the center and report back some statistics, and then adjust the positions of the stage to optimally hit each well. So, and then last but not least, on the beam line, I want to emphasize that one of the things, sample preparation is important. There's some things that they're unstable. You have to manipulate samples either right before or right after. And NSLS2 is a big facility. It's an 800-meter circumference storage ring. Even though we have labs close by, they're still, 100 steps from the beam line to the lab. So if you're doing something that has to be done quickly, running back and forth between the beam line and the lab is going to get old quick. So we built a partitioned area at the beam line that allows us to, users in particular, ample bench space. We have a centrifuge, we have a plate reader, which we use for high throughput Alexa dose response characterization, a variety of other, gizmos to do experimental work. And then I should emphasize that more complex work, such as cell culture or purification of samples, their new structural biology wet labs just around the corner from the beam line are available. So I've said a little bit about the beam line. I'm going to say a little bit now about how you get access to beam time at NSLS2. So, I think I'm going to hit on some high points, but quite honestly, there's a NSLS2 user guide that's really well written, gives you step-by-step instructions on what to do. I have a link here, but so just kind of for reference, NSLS2 operates in 3-4 month cycles per year. All access is via peer-reviewed beam time proposals at no cost to you, with the exception of proprietary work. And I show the cycles and the typical proposal deadline, which is about 3 to 4 months before the cycle in question. And just to kind of reiterate here, especially if you're planning for pretty much any experiment, even if it's just remote, your institution should have a user agreement with Brookhaven Lab that governs your access to the user facilities. You need a guest appointment to access the site and facilities, and this quite honestly is true even for a purely remote access or mail-in experiment. You still should have it. And there's also a risk assessment process with samples, and for most things they're non-hazardous, not really a big deal, but if you're doing, for example, BSL2 materials, much more close scrutiny of your what it is and how you're going to handle it. Do you have the appropriate level of knowledge? So proposal type. So we have quite a few different types of proposals. I would be happy to discuss specifics of what works for everyone, but sort of just to highlight a few things. The standard general user proposals, they're accepted 3 times a year and they last for 1 year. And you can request up to 3 beam lines on a single general user proposal. So you can propose, for example, a study that would combine solution scattering and footprinting and maybe crystallography in a single proposal. Rapid access, that's rapid turnaround, and the goal is within 2 weeks of submission. And that could come in at any time. That only lasts for a single cycle, and it's only 1 beam line. One thing that we have at NSLS2 that's a little unique that was imported from Europe is an idea of what's called a block allocation group. So that's a group of geographically or scientifically connected investigators who require guaranteed time. And these uniquely last for 2 years, up to 6 cycles with mid-tenure review. And more or less, if you get a favorable score, you're guaranteed beam time on the beam line. And it's up to you to organize how you want to use it amongst the various participants in the bag. And these allow up to 5 beam lines to be selected. So again, we're very interested in trying to push multimodal, multi-technique, multi-beam line approaches. Then of course, proprietary users. So this, as a note, anyone who requires control of intellectual property and for proprietary reasons can submit a proposal that way, that you are charged for beam time at a not insignificant rate. These, however, can last up to 5 years, and it's very flexible in terms of number of beam lines, and we will try to make the access process as easy as possible. I sort of want to close by pointing out that, consider the objectives and scope of your program when selecting your non-proprietary type, because, the durations and the nature of them vary a lot. So my last slide on access. So for XFP, we love having on-site users, and that's particularly true for new users. Introduce them to the beam line, show them how to do things, help them think about the experiment. I mean, I have to say in some ways, there's no substitute for doing it yourself. It is required for certain types of experiments. So if you want to do BSL-2 work, you must be qualified to do that, and you must come on site to do the experiment. But we do support a lot of mail-in and remote, particularly, if you're an experienced user and you've been here and you know what you're doing. Currently, I want to emphasize, though, that we have no on-site user access due to COVID-19. So all experiments for the rest of the summer, at least, and possibly longer, will be mail-in on XFP, and that's general to NSLS2. So that's all other structural biology beam lines as well. So go to highlight some thoughts here that I want to emphasize on, what goes into a successful experiment and a proposal. The big thing, as Corie said, is, come and talk to us, contact us. What do you want to do? We can help you, think about the experiment design, whether it's feasible. Think about, understand your goals, and it is required for certain experiment types. If you want to do a BSL-2 experiment or an in vivo experiment, you have to contact me to talk about it. And the other consideration, I think, is to think a little bit about your access to and experience with mass spec instruments and analysis tools. I mean, this workshop is one way that we're trying to build up the user community to do that, but not everyone has that. So if I understand your what you know and how your ability to actually analyze the data on exposed sample, that I can help find ways to increase your success. I mean, the worst thing is to have someone come in, expose samples, and then be unable to analyze them. And lastly, and I just kind of I throw this out here every time, be aware of the support labs and the facilities that we have. So there's a lot here, but also be aware of the safety culture at national labs. Some things get a lot of scrutiny, and you may not get away with things here that you get away at other places. In closing, Beamline is open to users. I'm happy to chat about feasibility, access, projects. If you're in a position when the facility reopens to visit NSLS2, by all means, stop by XFP and say hello. And I want to take this one point to actually, if you're looking for a job, we're hiring. So we have a postdoc position open, focusing on science projects and technology development at XFP with a bit of user support. You'll be based full-time at NSLS2, and please feel free to contact me about it and we can talk more. And I would be happy to take any questions at this time.
Great. Thank you, Eric. Eric, I realized I forgot to introduce you. I'm really sorry.
Yeah, that's why I kind of had that odd pause, but.
So Dr. Eric Farquhar is, as you've seen, an expert in X-ray footprinting, but I just wanted to point out he's also an expert on X-ray absorption spectroscopy. So if you have other questions about spectroscopy, you can ask him that as well. So of course, if you want to do some footprinting experiments, reach out to Eric or reach out to me. and we will help you get started on either the East Coast or the West Coast footprinting beamlines. So I'm going to actually enforce a break right now. And I'm sorry, Sinjin, I know you're next, and I hope your time allows for this. But let's actually take a Zoom break and reconvene at, let's see, it's 11:07 right now. Well, let's reconvene at 11:10. So that gives us a 3-minute break.
All right, we'll be sprinting.
Everyone sprint to the bathroom and come back. I'll see you in 3 minutes. And we're back. Sinjin, are you there?
Indeed, I am.
All right. So I'm really pleased that Sinjin Skilton can talk with us today. So he's worked at Protein Metrics for 5 years. Before that was at SciAx and Waters, where he worked on mass spectrometry development applications. And of relevance to today's meeting, Sinjin has worked on commercializing HDX software. So if you are interested in HDX, he is really the person to talk to. But now he's going to tell us about Protein Metrics software in general.
Thank you, Corie. And yes, I'm probably the most boring of the people you get to listen to today, so I won't bore you for long. But I should also mention at the same time that one of the people that is on this call, who's one of our colleagues, Ilka Sen, is also a very highly respected expert in HDX and has worked quite a lot on a lot of the oxidative footprinting stuff as well. So I'll be very brief. Realistically, what we are is in essence a tool for many of you guys, in the sense that you end up getting huge amounts of data to process, and we've got to help you process that data. So what we've done is we've developed a whole set of workflows that are based around higher order structure. For those of you who don't know Protein Metrics, we have a platform called BIOS, which automates a lot of workflows, many of them geared towards biopharmaceutical or protein characterization work. I won't dwell too long on that, but we're very well established in both the biopharmaceutical and academic worlds for this type of work, and we're highly focused in this world. Of relevance for today is that we have workflows that are geared specifically towards higher order structure. So obviously oxidative footprinting, which we've got a little FPOP label in there, and then also hydrogen deuterium exchange, which is currently in beta. So we see that as part of a higher order package overall. There are lots of reasons to do it, not just some of the work you're seeing here today, but also because, in fact, it's highly demanded by the regulators in the biopharmaceutical world. So they specifically ask for this as part of the guidance. And it's always a bit of mystery to us why oxidative footprinting and radical footprinting and those techniques aren't much, much more prevalent in this world, because they tell you so much. They tell you so much more information than you would otherwise get from some of the older techniques as well. Specifically, when they went into a bit of head scratching back in 2009 to 2014, eventually, in going through, well, what exactly should we be doing with biotherapeutics? They realized, well, yes, we do really need to have this as part of footprinting the molecules overall. And it became, much more obvious to people in the FDA that three-dimensional structure really, really had to be properly understood. Inside industry, of course, it's absolutely critical, because it gives you a whole lot more control of your processes if you have the right overall three-dimensional structures. Very, very importantly, and this has been in the news recently with IP on many of the molecules that go into biotherapeutics, sometimes it's about their structure. It's not necessarily about the sequence. And of course, they need to protect IP from that point of view. And in the end, of course, something which affects every single one of us here, ending up with higher quality biotherapeutics when you control these processes better. So this is one of the reasons why these higher order structure tools really, really are quite critical. I'll go through some examples that we've worked on in the past years. So disulfide bonds obviously is another area too, which is really secondary structure, not tertiary structure. But of course, having that controlled is really critical for all the reasons above. And we did a study a few years ago where we looked at this for some biosimilars. If you're delving into quaternary structures, some of the work we've done with Stanford in recent years is basically looking at how things are all stuck together, which is quaternary structure, and that's another application of this. Obviously, we've done epitope mapping, and this is one of the posters we did with Michael Brenovitz, where we're trying to do it on the benchtop. I think you'll hear a bit more about that in the future. And then, of course, the hydrogen deuterium exchange. And one thing I want to call out here is the kick in the pants that we were given many years ago by Michael Gross, who said, you better start organizing this stuff so that we can process it properly. And of course, being able to look at HDX data and footprinting data at the same time is pretty critical for a lot of places too, because they might be complementary in information. What we've done as an HDX software is really an extension of some of the other platforms we've got. So it gives the people access to the data itself as well as some of the results, so uptake plots and things like that. But also more critically, I think for interpretation of this, we've got reporting structures where you can do things like the wood plots, and Corie had mentioned earlier being able to look at some of the crystallography data and NMR data. Well, you can actually bring in a PDB, a ribbon plot into our reports and map your changes onto that. And in a nod to what some of you guys were saying, well, this is actually cytochrome C, very poorly mapped. So as Lisa commented, it's not exactly a straightforward one. Some of the articles that we've done in the past, so Genentech obviously took on our software quite early on in the process of having it commercialized, and did a number of designs. And they, of interest, of course, to us was this was in antibiotics, and being able to look at some of these things and how these lipopolysaccharides actually work. So that was pretty interesting for us. Going back to the Fenton chemistry that was mentioned earlier, of course, this has got some legs, if you'll excuse the pun, in terms of being able to do it in 96-well plates, so actually making it a lot easier to perform the radical footprinting. And then, of course, the typical way in which you might be using this for a lot of people who are doing IP and all sorts of other reasons is, of course, which amino acids are protected in solvents and so on. So it's an interfacing between proteins and finding exactly how they interface. So epitope mapping, of course, is another one. Again, quite typical. And here, of course, it's going to be what's critical for us to be able to work through the data with is that this is not necessarily static pictures, but of course they're very dynamic. So being able to cope with all of these dynamic complexes is really, really critical. That's about it from me. You'll see so those of you who've signed up to actually get some of the software from us, you'll get a chance to quiz us about it. And Ilka will be able to talk you through some of the actual practical applications. And I think from Lena, you'll see some of the real world work that's been done with it. And all I will say is thank you for those of you who nag us to do something better, because generally we're making tools that should be useful for you, not just created in a vacuum.
Excellent. Thank you, Sinjin. So let's let us forge ahead. We're heading into the practical session now. So if we could have Ilker share your screen. And Ilker will give just a little brief overview of PMI for our particular application, and then we'll go into Lena's talk.
All right. Hi, everyone. So my name is Ilker San. I'm Customer Success Scientist at Proteometrics, and I'm going to be talking about our software. And I don't have any slides, so it's going to be live, hopefully slightly exciting, but we're going to keep it short. So we'll move on to the real world stuff of Linux. Okay, so some of you have requested, some of you have our software already, some of you have requested a demo license, which we provided. I think Corie and Lena also provided you some data files that you can try with. So this is part of the practical session. If you look at our software, you'll see several icons on your desktop, but the one you should be clicking is BIOS. So this is our workflow launching platform, and it has a list of several workflows that are shipped with the software. These are they include default parameters that we expect users to actually modify and then save as a part of their work, as a part of their own work. So just a brief outline of what is what here. So we have a bunch of intact workflows here at the beginning. These are for charge deconvolution on MS1 data. And they have antibody shapes, but obviously they support all kinds of intact data, including native, large molecules, really large molecules, and all that stuff. The next bunch here from PTM all the way to MAM reference characterization, these are all peptide-related workflows. and they typically do an MSMS search and follow that with quantification. And that is followed by reporting, where you can compare samples across each other, see how it changes. And this is what we're going to look at today, mainly specifically to the oxidative footprinting workflow. After that, we have actually a bunch of chromatogram analysis workflows. They look at either TIC, UV, or fluorescence chromatograms, or electroprograms if you have them. They can look at under peaks. If you have mass spec data, you can combine them, and they do those kind of stuff. A bunch of 3 detached glycans workflows down here. These are for usually released glycan analysis. And finally, de novo sequencing. Okay, so back to the meat. So we're going to be looking at the oxidative footprinting workflow, which I have modified and saved a copy, and that's what we distribute to this site, the oxidative footprinting. So let's take a look in there, and then I'm going to talk to you a little bit about what is in here, and then I'll show you how to get it started and what the results will look like. All right, so when you launch a workflow, any workflow in BIOS, you're going to be presented with a couple of tabs here. One of them is the samples tab. This is where you drag and drop your raw data files. Sequences, in this case, I've already added the cytochrome C sequence here, but if you have a FASTA file, this is where you would bring it. And then processing nodes is where the actual search parameters, etc., can be found. So this depends on the workflow, how it looks. So this particular one has a single bionic workflow, which does the MSMS searches, and it follows it with a biologic node, which does the quantification. So if you look at the bionic node parameters, most of these things should look familiar to those of you who do mass spec analysis. They will have things like, mass tolerances for precursors and fragments. What kind of fragmentations do you have? Where you want to add decoys to your FASTA database, your digesting enzymes. Down here is where things are very specialized for oxidative footprinting experiments specifically, and that is the modifications. So in this set, we have, for example, a list of oxidation modifications on a lot of different residues. And these are customizable, of course, depending on the experiment you do, what residues do you expect to be oxidized. And we have seen a quite variety of these today, which is exciting. Bionic, our software counts a bunch of glycan databases. So if you analyze any glycosylated proteins, you can choose one of the built-in databases to do those searches. And finally, there's nothing else down here really interesting or in the biologic node that are different. So in our case, we have how many are there here? 6 data sets here. One of them has MSMS. The other ones are MS2 only. And Lina and I have done this a little differently. What I did was to build a workflow that will analyze all of this at once. So what we'll do is just drag and bring all of those here into the samples tab. Let's make sure everything has copied in. Okay. And now what we're going to also do is to direct our bionic search node, the MSMS search, to only search the first file, so it doesn't try to do MSMS searches on the MS1 only files, because that would not go well. And that we do here in the samples tab. So we basically take what is in the file name, MS2, put wildcards around it. Anything that matches that is going to be included in the search. And in the biologic node, we have a complete wildcard, which means everything, all of those data files will be in the final project. And that's pretty much it. That's all you have to do here. You fire up the workflow we provided, you bring the raw data files, and then we create the project. That's it. If you have any questions, and then you'll save this. And then this is going to be like a cooking show. It takes about 5 minutes for this project. It's not long, but I'll launch ahead anyway. BIOS is going to present you with 2 tabs. One of them is going to look like this. This is our inspection view. And the other one is going to be the report. We're going to get to that in a little bit. Now, if you have any questions, start typing them up in the chat so we can answer at the end of the practical session and launch all the all the points that you're going to be raising. Okay, so what we'll see here is a big table of peptides. So this is all identified peptides with a lot of information about those peptides. Things are organized by unique peptides, which are defined by sequence, modification, charge state, and sample. So sample meaning the raw data file. Initially, we're not going to see any data coming from the MS1 only files, but only what's in the MS-MS identification. So we click on a peptide, and we're going to see here the extracted ion chromatogram. On the top right is going to be the MS-MS spectrum, and here is the MS1 spectrum. So this particular data happens to be centroided. That's why we see centroid. If it's profile, we would have seen profile data. If you click on a modified peptide like this one, we will see a divided panel view. So on the top is going to be the modified peptide, in this case oxidation. The bottom one is going to be the corresponding wild type. So this is useful for direct comparison in both MS-MS, MS1, and the XIC. Particularly useful in XIC because important in oxidation is sometimes you can have in-source oxidation, which is not coming from your sample, but it's coming from your process. And if you ever spot those, you can exclude them from integration by moving the integration bars. So we don't really have that here, so we don't. This data set doesn't have that problem. But if you have something UV inline or specifically high voltage at the electrospray, you may actually see those. And this will be the very easy way to spot. There's going to be these little dots on the XIC. So these are individual MS-MS IDs that identify that peptide at that exact retention time. And they may be regularly spaced like this. This just is the dynamic exclusion interval, basically. This just tells me what the dynamic exclusion is in the data. So that's expected. And we can click on those to actually jump on to that particular scan and look at that specific MS-MS. All right, so what we're gonna do here, so not much. We have to look at some peptides that have multiple oxidations, like this one here. And we have to make sure that the XIC peaks are integrated properly. And the reason why we need to do that is a lot of times you will notice, in especially oxidized peptides, come on several peaks, right? Sometimes the same residue oxidized will elute at different peaks. Sometimes it's going to be a different residue. And we can tell that by looking at these gray dots instead of the red or pink. That means it's the same precursor ion, but a different modification position. When we hover over it, it kind of tells us where it is. The lowercase tyrosine there is the modification position. So that's the other peptide. So we have to make sure the integration limits are set properly, that we're not integrating both of those, because it's the same XIC, right? We don't want to integrate both of them. We want to only integrate the peaks corresponding to this tyrosine oxidation. And in the methionine oxidation, we want to only integrate this peak, which is the methionine oxidation, and exclude the tyrosine oxidation. Most of, you don't have to do this for every peptide. The software does its best to hone in the XIC to basically include just that identification and peak. But if the peaks are too sharp, sometimes if there's noise in between, the software can make mistakes. All right. So we'll do that for, we'll do our inspection for all of those peptides that we want to look at. You may come across a peptide which you don't trust at all. And for those, you can go to the validate column and mark them as true positive, false positive, or uncertain, one of those. And finally, you can actually use those to apply filters. You can filter those things that you have marked and validated. So these three columns here are interactive: validate, comments, and labels. But everything else in the software is not, so you cannot delete or change any of the other values in this table. Okay. Finally, once all that's done, what we're going to do is we're going to add the XICs from the missing samples, from the MS1 only samples. And that's a single click thing. We go to edit, in silico peptides, and add missing via existing. So what this does is it's going to add what we call in silico peptides, which are basically peptides that don't have an MS-MS ID, for IDs that we already have in our project, but are missing XICs in other samples. So this will harmonize your project to make sure that you have XIC for every peptide in every sample. And that will also take like 3 minutes, approximately 3 to 5 minutes. So we're going to jump onto that, which is this project. So now each row, when you expand on it, you're going to see 6 sub rows, and these will be the individual samples. How it looks like in different samples, the same peptide. All right. So at this point, we don't have to do anything. We just, if the retention times are aligned, there is absolutely no problem. Which in this data is perfectly aligned. Everything should match up, and we can move on to the report where we're going to actually see the overview of these modifications across the 6 samples we have and how they differ, what are changes. So Lina will talk more, I think, about the sample and what we did. So I'm just going to very briefly mention it. This is, I think, different. Actually, I forgot what it is. I think it's different excitation time. Yeah, anyway, I forgot. We'll ask the expert Lina. But oxidation levels are different across samples, and this is how we can see it. We look at the report. This is a heat map view that shows every peptide identified. What are the modifications? They can be oxidation or sometimes dioxidation, which residue, and then what is the percentage of that modification across the 6 samples that we have here. So we can see here clearly increasing across from the first sample to the last. So this is one kind of view. Another view is, this is the default report, by the way. Another view here is, again, percent modification, average across different chart states this time, but has these little bars in the tables that show you trends within the same row, which is useful in seeing, if something is going up or down. Finally, you can make the same report look like a bar chart or average across different residues. So this is in the default report. Again, it actually shows you the average modification found in a particular residue type in the sample. So this largest one here is methionine, which is expected. And the average methionine modification is different in each sample, and it changes. So this is not site specific, but looking at the all methionines at once, particularly useful. I didn't plug in a PDB file here, but you can, and you can map this kind of data on a PDB file as Sinjan showed. And we can customize all this using the configuration and apply filters, etc. So we're not going to go into that much detail here. Just keep those in mind that those are possible. And if you have any questions, just feel free to ask us. Okay, I think that's it from my side. Just a brief overview of live software, and then I'll hand it back over.
Great, thank you, Ilker. That was great. So I would like to now introduce Lina Christensen. So Lina is a new scientist in my group. She has a master's in biochemistry. And even though she only started at Berkeley Lab about 6 months ago, I would say she is rapidly becoming our resident expert in analyzing footprinting data, especially using the PMI software. So she's going to show us a video showing specifically how to use the PMI software to generate our standard dose response curve using some test data. All right, so Lina, I'll hand it over to you.
Okay. Yeah, I just, before I play the video, I just want to say it's going to be a bit of a repeat of what Ilker said, so I hope you don't mind getting it twice. But I'm just going to go ahead and play that video.
And I'll just say, I think it's totally fine to repeat some of this information. It's hard, it's complicated software, so this is good.
I will be giving a demo of how we use the PMI software in the analysis of LC-MS data from X-ray footprinting experiments. We'll be looking at data from the X-ray radiolysis of samples containing cytochrome C, which was the protein used to test the JET sample delivery system at the ALS footprinting beamline. Before I start the demo, I would like to give a brief overview of the 2 workflows we use in BIOS. The data analysis process I'm sharing with you today has worked well for us, but I should point out that there are several ways of working with in silico peptides in BIOS. In the cytochrome C example, we're analyzing one moderate exposure MS2 file in the oxidative footprinting workflow, which is where the identified modifications are scored and validated prior to the creation of an in silico peptide library. After the creation of the in silico CSV file based on MS2 data, the in silico file is then applied to several cytochrome C samples in the PTM in silico workflow for which mass spec data was acquired in MS1 mode. This combination of MS2 and MS1 data ensures that we can identify residue-specific oxidative modifications while at the same time achieve good data quality. The final step of the analysis involves the creation of dose-response plots in Excel, from which we derive the rate constants. So let's take a look at the data. Here we have the overview of the BIOS workflows, and we're going to go ahead and select the oxidative footprinting workflow. And I will launch a workflow that's already been set up for cytochrome C. The workflows consist of 3 tabs: the Samples tab, the Sequences tab, and the Processing Nodes tab. In the Samples tab, you'll be bringing in the mass spec files that you'll be working with. And then in the Sequences tab, you'll bring in the FASTA file for the protein or proteins of interest for your project. And then in the processing nodes, there are several parameters and settings. And I'm not going to go over all of them in the demo today. I just want to highlight a few. One is for the digestion. Right here we have specified a trypsin digest, and we're allowing for 2 miscleavages. And then in terms of the modifications, we have specified fixed and variable modifications that are appropriate for an oxidative footprinting workflow. And then once you are satisfied with your parameters, you will go ahead and click the button down here, Create Project, and that will start the project creation. Once the project has been created, you will get a report with several tabs. And one of the first is the PTM summary tab, which first of all shows you the degree of sequence coverage. In this case, we're at about 80%. And then it also gives a really nice global view of the degree of oxidative modifications that have been identified for the peptides that have been picked up in this particular analysis. In the peptides table of the project, we have access to all the peptides identified from the MS data, both native and modified peptides. When you select a peptide with a modification, you will see the associated native peptide down here in the wild type peptides table. And further down, we have the extracted ion chromatogram plots. The top panel shows the XIC for the modified peptide, and the bottom panel shows the XIC for the native peptide. Over here to the right, we have the MS2 fragment ion spectra, and you can also view that data in a table view format, which is very helpful. And then finally here in the bottom right part of the screen, we have the associated isotope plots. In terms of validation, what I like to do first is to take a look at the scores and the delta mod scores across the entire data set. I like to just kind of go through and get a feel for how good the data quality is as reflected in the scores. Another thing that's really helpful is the ability to filter the data based on filters set for specific columns. For example, if we're interested in looking closer at the scores, we could go ahead and filter for peptides scoring, for example, below 200. So we have a few peptides here that are scoring on the lower end. And it's really nice just to have the ability to focus on, for example, low-scoring peptides that might require a bit more attention in terms of validation. And once you've used that filter, you can go ahead and remove it or apply other filters. Another feature we look at as part of the validation process is the retention time of the modified peptide versus the retention time of the native peptide. And we typically see a negative peak shift for peptides with an oxidation modification. So that's another good high-level check as part of the validation process to make sure you're actually seeing that expected shift in retention time. If we take a look at a couple of the modifications in this particular data set, we have a peptide right here that has two modifications, a tyrosine modification and a methionine modification. And what actually happened when this this project was originally created, this was reported as a mixed modification, meaning that the XIC area included all the peaks shown here, basically encompassing both modifications. I took a look at the retention times for the tyrosine modification. We have two peptides associated with that modification, and then for the methionine modification, we also have two peptides. And it so happens that the two tyrosine modifications are associated with the two peaks right here, whereas the two peptides with the methionine modification are associated with a peak right here. So I went ahead and split the XIC area into two areas that are more appropriate for the tyrosine and methionine modification. If you have a situation where your retention times overlap in terms of a peptide that has multiple modifications, you can just go ahead and report that peptide as a mixed modification peptide. Let's look at another peptide modification. Here we have a small peptide with a lysine modification, and 3 peptides were identified for this particular modification. The first 2 peptides are associated with the first peak right here. However, the third peptide has a retention time that puts it in the second peak here, which is a little suspicious. If we look at the fragment ions that were identified for the third peptide, we see that we don't have great coverage for the glutamine and the lysine residue. And we can also take a look at that in the table format. As a result, I went ahead and labeled that particular peptide as uncertain using the validate column here. In this column, we have 4 choices. We can leave the modification as unassigned, true positive, false positive, or uncertain. Since we have this third peptide labeled as uncertain, we're also going to go ahead and adjust the XIC area so that we end up with a more appropriate XIC area for the 2 remaining peptides with the lysine modification that we have labeled as true positive. Once you're satisfied with the manual validation, you can go ahead and set the peptide filter that will be used to generate the in silico CSV file. Here I've set the minimum score to 200, and we want to make sure we deselect the peptides that have been labeled false positive and uncertain, since we don't want those included in the CSV file. And then we'll go ahead and click OK. And after that filter has been applied, we can see that in our validate column, we're left with peptides that are either unassigned or those that have been labeled as true positive peptides. So at this point, we can go ahead and generate the in silico peptide library, and we do that by going to the edit menu and in silico peptides, and then we export in silico to CSV. Once the in silico CSV has been created, we can go ahead and launch the PTM Pure in silico workflow. Here in the samples tab, we have the MS1 files that we're going to apply the in silico CSV to. And then in the sequences tab, we have the same FASTA file for our protein of interest that we used in the oxidative footprinting workflow. And in actionable nodes, we're going to go into in silico options and make sure that for the in silico peptide CSV, that we are pointing to the CSV file that we just created. And once that has been set up, you would go ahead and create the project. And so here we have the result of the pure in silico PTM workflow. We have our 5 MS1 samples, and we can see here we have that nice global view of the peptides that have been identified with the associated modifications. And then if we go to the second tab, we have a pivot table with all 5 samples listed here in the top row, and they are listed in order of increasing exposure time. And then in terms of the column fields that have been brought in, we have the type of modification here. It's dioxidation and oxidation, and we are also looking at the specific residue modification in terms of the position number and the amino acid that has been modified. And if we could just kind of quickly take a look at the data here, we see a general trend that as the exposure time increases, we see an increase in oxidation as expected. At this point, we could look at a couple of specific peptides. What I did is that I made a copy of this tab, percent mod by mod name across samples. I went ahead and made a copy of that tab here. And then I selected for 2 specific peptides. One has a tyrosine modification, the other one has a methionine modification. And these are the 2 peptides and the 2 modifications we're going to use to create dose response plots in Excel for this demo. And I just wanted to show that you have a lot of flexibility with these these pivot tables in terms of bringing in the column fields that you're interested in. So what I did here, I brought in the sequence and I selected for the 2 peptides that I'm interested in. And that's how I was able to narrow it down just to these 2 modifications. And so the data we have here is the percent modification for various exposure times for just these 2 modifications. From here, we can go ahead and export this data to Excel, and we do that as an export pivot table content CSV. Once you export the pivot table content to Excel, you end up with a CSV file that looks like this. And I took the data and I transposed it into a format that's a little bit more conducive to generating dose response plots. So we have the original percent modification figures here for the tyrosine 97 and methionine 65 modification. And I went ahead and calculated a fraction unmodified on the basis of those figures. And I also normalized the data for the methionine modification, since we do have a little bit more than 2% background oxidation for methionine The normalization of the data basically ensures that the dose response plots are reflective of just oxidation related to footprinting and not background oxidation. And so once you have your fraction unmodified data calculated, you can go ahead and plot fraction unmodified as a function of exposure time. And on the basis of that plot, we can go ahead and generate a trend line. This is a single exponential fit. So we have one here for each residue, along with the associated trend line equation and the correlation coefficient. From the equation, we can derive the hydroxyl radical reactivity rate constant, and I've shown that down here for both residues. And the rate constant is a measure of both the intrinsic reactivity and the solvent accessibility for that particular residue. That's all I have today. Thanks for listening, and please let us know if you have any questions. Thanks.
Great, thank you so much, Lena. That was terrific. All right, so this is now the part of the workshop where it is wide open for questions for anybody. And I have the power to put people in breakout rooms. So if you have, for instance, your PMI software up and you want to talk to one of the experts online here, just let me know. And you want to share your screen and talk to them directly, we can set that up. So I'm just gonna open it up to questions about any aspect of footprinting. I see there's a number of questions in the chat window that have been, I think, maybe answered.
Yes, I think between us, we answered some of them.
Yes. Oh, and to the question about, is this presentation that Lena just gave available? Yes. So I'm going to post it actually to our footprinting website. I'll just type that web link here again, so you have that. So in a couple days, that'll be up.
Of course, we should also say at the same time that we're aware that almost every single data set is different. So don't feel shy about asking us. We do that all the time, help people with data sets.
All right, I just put that website up in the chat window. So my hope is that over time, we're going to post more and more instructional videos up there to help people out with the footprinting method.
I have a question, if that's okay.
Sure, go ahead.
Okay, with the novel CF3 labeling chemistry, I sort of took from it that CF3 labeled that proximal histidine, whereas the OH radical did not. And I've been reading that proximal pocket is considered very hydrophobic. There's a paper waterproofing the heme pocket. So I'm kind of interested in thoughts. Was that maybe only the apomyoglobin form, or was it also the holo? And if so, what the thought is on that?
Jana, I think you're
mute. Okay, yeah, I have to unmute myself. Sorry, the question was, did we also see this labeling in holo and upper forms, right? Am I understanding correctly?
Yeah, specifically for that proximal histidine.
You mean that three histidines were labeled there, right? Not just one.
Yeah, because, but I'm really interested in the one on the F helix. I think it's the H92 about. That one specifically is in that proximal pocket.
Yes, we've seen this in both forms, and that's why we calculate the difference by taking the ratios. So we observe this labeling in aper and a holo form, but in holo form we observe just decrease of labeling. It was much less than in upper form.
Why do you think the CF3 radical would modify but not the OH radical?
OH may be modified too. Like, I mean, if you remember one of my slides, I show that we have some histidine labeled by hydroxyl radical, but it's quite weak usually. So, and if and CF3 actually histidine is really very reactive. So you kind of have a majority of residues labeled are histidines and tryptophan, so you just see more of it. So that's why you can quantify easily. And in terms of hydroxyl radical, it was probably in the background. So we just cannot separate it and detect it. So, but because it's just low intensity.
Okay. Yeah, thank you.
So, and again, the chemistry might be slightly different, so we don't know yet. So we're trying to kind of learn about like different differences in chemistry, how we label with CF3 and OH like basically at the same time, because because we see different patterns obviously. So, and we need to make sure how we're gonna titrate this chemistry as well to see different modifications. So this is still an open question and we're still learning this. So we're doing a lot of experiments now to understand reactivity, solvent accessibility with CF3. So I think that as we go, we're gonna have more answers. So this was just the first analysis on with known structure protein, like using synchrotron just to see if we can label actually and like evaluate the pattern of labeling.
Okay, thank you.
Just looking back through the chat, it looks like Cyan had a question about the CF3 labeling as well. Cyan, do you want to ask your question?
Yeah, so can it cancel each other, that OH labeling and CF3, that you have one site in one conformation, the OH is showing protection, on the other hand, CF3 is showing more activity?
You mean going in different directions?
Yeah.
We haven't seen this, but again, we have a very small yet set of data to, to compare. We're looking at the peptides right now. So it seems like the labeling goes, at least on a peptide level, like if we label with CF3 and we label with hydroxyl radical both. So we still see kind of in the same direction. But again, I cannot really tell right now because we have such a small sets of proteins we tested, so then we can really definitely say that this is the case. So what we tested so far, it seems like direction is similar, at least for overlapping residues. So the residues which we have like labeled differently, so obviously we have a different information. But overlapping residues were behaving on myoglobin in a similar way. So we had an increase in CF3 labeling, we saw increase in hydroxyl radical label as well, versus in two different forms. But again, it's a very small data set, so we can't really yet tell everything about this chemistry.
I see.
Yeah.
Thank you, Jara.
Yeah, we just learned that solvent accessible, react at its act, we label as solvent accessible, and we have some residues we label with both, some residues we label with the different chemistries in the same sample.
Thanks for the explanation.
So it looks like we have a question in the chat about the normalization of footprinting data, generating the dose response. A really good question about, here, I'll just read it. So if each peptide is compared to its parent unmodified in each sample, does that make it such that the differences in sample injection amount won't matter? So that's a great question. We have some experts here who can answer that, Cyan or Lena or Jana. Anyone want to?
I think if you do normalization, your sample loading will not matter. That is correct, right?
It will if you have a difference, let's say 10-folds. So you would see that the fraction of modified and unmodified would be affected because one is because of just simple dynamic range and mass spectrometry. So you have low abundant species versus high abundant. So that's why you don't want to go this low abundant goes into noise, because when it goes into noise, you just can't quantify properly. So, and that's why you start to see more effect if somehow you load in much less material for one sample and the other. We're trying to make sure that we on the same, like we load in the same amount. So we kind of normalize the amount of material we load in for like all experiments, just to avoid this kind of dynamic issue and effect on the peak area quantification.
But still, you should see it will not be affected, the overall rate.
It depends again how low you go into low abundant, because if you like 2-fold difference, there is not much effect. But if you go in a 10-fold difference, you start to see dramatic effect in you completely change the rate of modification literally. So because of this issue. So because again, you have to be cautious because we're doing mass spec to analyze and quantify peak area. Every time you're in a noise, so you start integrating in different things, so which can affect and change in a modification form a little bit dramatically will change your modification rate. Change in unmodified doesn't a little change in unmodified doesn't really affect much overall fraction of unmodified. So the modification is very important. And when you start to change in this peak shape and getting into noise, you affecting a lot your modification rate at the end. And I've seen it on many, many samples, especially when you do pepsin digestion, because you have a various amount of labeling. And again, amount of like, you digest peptides in a different way. It's not 100% of each species. You have a multiple species present and a different volume. So it's mean that you start to see a lot of effects because of this when you don't load a similar amount of protein for each experiment.
Great, thank you. That's something to watch out for. I mean, I'll just say also, you know, sort of not really the same issue, but, you know, one of the things you have to be careful about in footprinting experiment as well is making sure that, I mean, when you're using X-rays, that you don't have too much modification of your proteins, because the problem is that if you are starting to modify a protein and then the fact that the modification affects the modification rate of either that residue or surrounding residues, that can really affect your data. So you have to really try to stay in that, what we call the linear range of the dose response, so that you aren't changing the rate of modification during the experiment. Okay, I have another note. Let's see in the chat window here. So if there are no other questions, I guess one thing we could do is we could go through how to load the data once again into the PMI software if people are interested in that. I don't know, but I know that some of you downloaded the demo and maybe are playing with data. I see a yes. So let's see, how should we do that? Ilker might be the best person for that if he is still on.
Yes. Okay, so how do we do it from scratch, or how do you do it from the workflow that we sent for this particular data set?
I think, Ilka, if we can do it, you know, because we send the FTP with the files and the workflow and the FASTA, I think if we could do it, you know, loading the files and then opening the workflow and then loading the files, I think that should be, they could replicate that.
Okay, sure.
Thank you.
So the easiest thing to do, you fire up biosoftware and then you go to File and Launch Workflow. With this window, you basically point to that particular workflow file, and that it has a WFLW extension. So that's in the email, right? And that will open up this window. It's going to look exactly like that. So this workflow we already set up so that it has the sequences, it has the correct parameters. So all you would need to do is bring in the raw data files. So I guess this answer is also just to demonstrate what we're drag and dropping here are the raw data folders for Agilent instruments, which have .t file extension. Bruker also has a .t file extension. Waters data files have .raw, and those are our folders, and Thermo has .raw as a singular file. Or if you have Shimatsu, I think it's an LCD file, something like that. Anyway, so you just choose them all, whatever you like, and it can be a mixture of instruments too, that's not a problem. You just drag and drop them here. The first tab, samples tab.
So Ilker, what if you don't have a workflow already set up?
If you don't have a workflow, you start from scratch. You either choose the PTM workflow or the oxidative footprinting workflow. They're very similar to each other. For particularly oxidative footprinting, you would probably want to do this because it has a list of modifications that is a little different than PTM. And same deal. So in the first tab, you drag and drop all your raw data files, just as the other one. This one doesn't have, of course, built-in FASTA files, so you bring in your FASTA file that has your proteins, however many there may be. So everyone except Lisa Jones, okay? Because Lisa is looking at thousands of proteins, not one or two. So Lisa will have to start from the HCP project. And that is configured to read, actually get a FASTA file as a whole, so you don't have to select individual proteins. But for all other experiments where you're looking at one, two, or a few proteins, this will work. And then in the processing node, so this is you're going to customize based on your instrument and experiment. Most is going to be the first tab. So here we're going to say which samples we want to run the MSMS identification on. Is it all samples that I dragged and dropped in, or some of the samples that match to a certain string, which is our case here. So, right, we have only one data file that has MSMS, this one. All others have MS1 only. So we don't want to run bionic searches on MS1 only data files. Do we want to add decoys? Yes, we don't need to change that. Precursor mass tolerance, this you change based on instrument. So we can, I think this was Agilent, so we said about 20 ppm to 40 ppm for fragment mass tolerances, something like that. It's a little wider than usual, but it shouldn't matter. Fragmentation type, again, depends on instrument. For any TOF instruments, this typically works QTOF/HCD. For Orbitrap HCDs, you can use this. If you have multiple fragmentations, like HCD, ETD, etc., you have some options here. Or if you have a Thermo instrument, you can choose this one, which just reads the scan header and then goes from there. So for this one, we're going to choose QTOF-HCD. We don't care about second fragment mass tolerance because we don't have two kinds of fragmentations. We only have CID in there, right? We're not recalibrating. You can, if you have lock mass, you can enter it here. This is triptych digest. So digestion settings are pretty standard for triptych. We're looking at fully specific. If you'd like to identify as much as possible, of course, you may want to change this to semi-specific, but in our purposes, typically not necessary. Modifications, yes, this is where we really need to optimize, and this is where Bionic is different than other search engines out there. So in Bionic, there are fixed modifications and there are variable modifications, but there are two types of variable modifications. And one type or another, common or rare, it doesn't matter too much, but the combinations of how you set the rest of the modifications changes what possibilities are there in the identifications. So for this, we have a bunch of documentation, application notes, or a few even videos that we talk about this modification fine control in Bionic. The idea here is to give user the maximum flexibility in setting variable modifications without increasing the search space exponentially, even if you do look for a large number of modifications. Which is what we do here, right? We have oxidation. We're looking at oxidation on several different residues, and you can keep adding to this. If you want to add alanine, just add alanine. But we are also looking at proteins that may have an end-term, pyroQ, and if that happens, it's typically around, almost 100%. So we want to make sure peptides identified with end-term pyroQ can also have an oxidation. So that combination is allowed. But we're not going to be identifying, for example, in this case, a dioxidation and an oxidation at the same time. We're going to be identifying one or the other on a peptide. Right, so that's typical modifications here, nothing groundbreaking. But if you look at some of the other workflows, you will see that they're going to be different depending on the workflow. And how many of those totals are allowed? So for each modification has a type, common or rare or fixed, and how many of those commons or rares are allowed in total. Okay, glycan databases. So there are N-linked glycan databases, all-linked glycan databases, and small and large of them that are actually shipped with the software. So you can choose any one of these depending on where your sample is coming from. For N-glycans, 50 common biantenary is the most common 50 ones found in biopharmaceuticals, but also other samples, tissue samples as well. But if you have plasma, you may want to change it to human plasma. That will look for N-linked. If you're searching for all-linked, then just choose all-linked. That's it. Okay, so don't typically change the rest of the settings here for oxidative labeling. For other kinds of searches, of course, you may. For example, if you're looking for glycans, somebody mentioned that, I guess, one of the questions. And if you're specifically after just glycan identifications, you may want to apply peak filters and run a wildcard search on glycans so that you can identify anything that is not in this database. But again, without expanding the search space indefinitely or to infinity. But for this experiment, we don't change that. Nothing else is, we don't change anything else here. I think just one thing to keep in mind, Bionic Search does apply automatic score cutoff, which means anything above 5% post-discovery rate will be actually filtered out of the results. So that's what that is. And in the Quan node, let's expand DOL. So there are options here that you can use in other analysis. For example, if you don't have MSMS data at all, you can just do it in silico digest here. Or if you have a list of peptides in a CSV file, you can import them here. That's not what we did for this particular study. We actually did search the MSMS file, and we added the in silico peptides based on the identifications made in that CSV file. So, sorry, that MSMS file. That's it. I guess we don't change anything else here. And then we created the project. Any questions about this process? Sorry, I'm not looking at chat.
I have a quick question. So, what do you convert data to MZXML format or any format, or you just use the raw data from different instruments? From any instrument.
That's right.
Okay. So then, how you is it software converted them itself, or what happened next?
Depends on vendors. So we typically have, for most vendors, we have collaborations with them, and we have their APIs to be able to read their data.
Okay.
So we use those. So we don't necessarily convert it. Bionic itself has a converter. We have our own data format. Sometimes in the background it happens, but it's nothing you see or even use later. Again, depending on the data and the vendors, is maybe different from vendor to vendor. But from a user perspective, all you do is drag and drop the raw data.
So actually we have, so just to clarify, we have formal agreements with all of the mass spec vendors to convert their data. So 99% of the time, it's the way in which they recommend it gets converted. Occasionally, we also have to chip in with converting stuff they don't do.
Does it process ion mobility data from broker?
Good question, Jana. So yes, there is specifically a column that reads out the Timms-Toff ion mobility value. So we don't use it for pre-discrimination, but you can use that number in the report to go and apply the discrimination by ion mobility.
Okay. Can you quantify then this data as well from ion mobility using the software or like?
Yeah, the quantification is exactly the same processes as for any other data. So, you know, giving all the caveats for quantification by mass spectrometry, of course, you know, that's exactly the same as anything else.
Thank you.
Okay, so I think we are winding down. Put some contact info in the chat window. So if anyone has additional questions after the workshop, you can reach out to people and ask them. I think this has been a really great session. We heard some wide-ranging talks about various advances in footprinting, which is really interesting. Learned about how footprinting works in general. And also we had a great practical session with some hands-on tutorials on how to actually analyze footprinting data. So really great to see all of you. Don't hesitate to reach out if you'd like. And hopefully we'll see you at the Beamlines, one of the Beamlines maybe in the future when we're actually allowed to go to bring users back on site post-pandemic. All right, I'm gonna end the meeting and sign off and hopefully talk to you guys soon.
Good luck getting on site.
Thank you.
Thank you.
Thank you.